So I'm just going to share my screen before we inaugurate the event. I hope you can see. So again, welcome everyone, whoever joined this Indo-UK Symposium on Fluxomics of Microbe and Plant Systems. So I first of all welcome and request our esteemed Professor Ajit Kumar Chaturvedi, who is Director IIT Mandi, to kindly inaugurate the symposium and as well as address the participants. Sir.
Thank you, Shyam.
Yeah, so I'm just stopping my sharing, sir, and please go ahead.
Thank you, Shyam. I'm delighted to be here on this occasion to inaugurate this international event, Indo-UK Symposium on Fluxomics of Microbe and Plant Systems. And I had not heard about this word earlier. It resonates similar to economics. I think there is some kind of balance that is taking place. If I look at the sessions and I was going to the website, and I saw that there is a flux balance analysis in plant systems and metabolic flux analysis in plant and microbial systems. So this looks like a very, very interesting topic, and I'm sure that it has relevance to both agriculture and also industry. And of course, it will take a systems biology view, as I understand. And on this occasion, I have a few things to share with the audience, with all the participants. First thing is that I'm very sad that we are not holding this workshop in IIT Mandi. So I wish that this workshop would have been held at IIT Mandi, and then all the participants, all our collaborators from University of Oxford and other universities from Europe, from North America, who are addressing and participating in this Indo-UK Symposium, they would have been able to see our very scenic campus. We are located in the state of Himachal Pradesh of India. It's the northern part of the country, and there we are situated in a town called Mandi. But it has come out to be a very beautiful campus in the Kamand Valley. So there are mountains around it, and it's a place that many people don't want to go back once they come there because it is so scenic. And I wish that this workshop, at least in future, if not this time, this kind of event can be held in the campus. IIT Mandi has now completed, almost going to complete 12 years now. And it has made a very niche presence in the entire Indian system as well as abroad, because it is very clear on its goals, on its vision. And the heartening feature is that this vision has a very important international component. And we are fortunate that we have got several colleagues, both young and senior, like Shyam is one of them, who are very energetic, who make sure that they set examples for others in terms of organizing events, in terms of doing good research, in terms of doing impactful projects. This is what we are trying to do. On this particular symposium, I would say that IIT Mandi is not divided into departments, but it is divided into schools. I mean, we do our academics and research in terms of schools, and this particular symposium will be of interest to not only the School of Basic Sciences, but possibly also of interest to the School of Computer and Electrical Engineering, because systems biology is an area that is getting increasing interest, presence from different people who have not done biology, who have not done basic science, who come from an engineering field. They also get attracted towards system biology. And especially when there are some quantitative methods involved, when there are analytical methods involved, or if there is an opportunity to apply data science. So I wish I could have stayed longer and participated in this particular symposium, but I don't think I'll be able to spend more time. I hope that the very galaxy of speakers that Shyam and his collaborator from University of Oxford, they have arranged a very impressive technical talk presentations over spread 2 days, and I understand that more than 150 participants are participating. I wish that all of them go back or get something very useful for their own career, for their own understanding from the talks. And these talks will be informative. I hope Shyam will take care that there is a good opportunity for interactions also, because this is one thing that will be missing in these kinds of online symposia, which is very easily, which comes naturally in the physical symposia, that we can easily mingle with each other, talk. This, I think organizers have to make special effort to make sure that this part is not missed out, so that it becomes more like also creating collaboration, creating partnerships by talking. So with these brief few words, I wish all the participants have a good time, and I hope that Shyam will also, and IIT Mandi will also gain a lot from this symposium. At the end, I would like to thank the Ministry of Education for having sponsored this project, Spark project, from which the funding for this event is coming. If it were not for that project, I'm sure this event may not have happened or may not have happened this year. But thanks to the funding that we got from the Ministry of Education in the form of Spark project, that we are doing it. I wish everyone a great time. Thank you, Shyam.
Yeah, thank you very much, sir. Indeed, it has been a great pleasure to get the workshop or the symposium, to be very specific, being inaugurated under your aegis. Thank you again. May I request IIT Mandi and Ken to give me the sharing rights so that I can share my screen. Yeah, thank you. So I'm just going to. So yeah, although this is a bit of a online symposium, still this picture reflects where exactly are we really located. This map is about all the Indian Institute of Technologies. Few more are yet to be added to the map, I believe. But we are in the northern part of India, north of India in the Himachal. I don't know if I can see my mouse or not. Yeah, and you can see we are nestled within the Himalayan mountains, the snow-filled ones, as well as both the campuses, north and south, very beautiful. And as Director Sir has rightly pointed, we are divided into different schools, and there's a lot of collaborative interactions among the schools. And we strive to scale the heights. So that is in a nutshell IIT Mandi. And whenever the opportunity does persist, we invite all the panelists as well as many of the attendees to go through this particular link, which has got the full book about IIT Mandi's journey in the last 10 years and all the programs, what kind of projects we are undertaking, and every aspect of that. So thank you very much. And before we start, I would briefly take this opportunity to introduce the symposium. This is Fluxomics of Microbe and Plant Systems. And we would be mainly aiming to explore what are the latest advances in investigating the plant and microbial metabolism. And for that, the themes are divided into day 1 and day 2, where on day 1, we would look into flux balance analysis in plant systems, that is today. And on day 2, we would be looking into the 13C metabolic flux analysis in plant and microbial systems. So the website link which is provided has further details. I encourage everyone to kindly go through that. So before I take any more time, I take this opportunity also to introduce our keynote speaker, Professor R. George Ratcliffe. He's the head of the department in the plant sciences at the University of Oxford. And to keep it very short, in fact, he's one of the pioneers wherein application of NMR to plant metabolic systems, as well as he's the pioneer in the Fluxomics, which eventually inspired several scholars, PhDs and postdocs, including myself, where I got an opportunity to do my PhD under him. And we are serving across the globe currently. So with this much of short introduction, I think I take this opportunity and my pleasure, indeed, it's a great pleasure to invite Professor George Ratcliffe to kindly give the keynote lecture of the symposium. I'll stop my sharing and then request George to start his talk.
We're starting a little ahead of schedule. Is that the plan?
Yeah, just yes, actually, yeah, we are ahead by about 10 minutes. If it is okay, there are several attendees already. We can start and then take maybe more time in question and answer section.
Okay. Right. Well, I'll share my screen. It doesn't seem to be sharing.
No, we can see your.
But it's not starting for some reason. Resume slideshow. Sorry, it's not starting at the moment. I'll just go out and start again.
Yes.
Right, so what I need now is, I seem to have come out of WebEx. Just a moment.
We can see you and hear to you, George. And
Can
the sharing
you?
rights are,
At the moment, I don't have your screen. One moment.
Yeah, no rush. We'll get this working. Yes.
I'm just coming back into the WebEx, I hope.
Yeah, we can. You are in WebEx. Yes.
Can you? I can't see you. I can't see anything. I have no WebEx screen.
Okay. Can you? Okay. Can you rejoin? May I ask the host to Chandrakant to kindly give me the sharing rights or take the sharing rights and then again give it back to George to see.
Okay, I'll give you the sharing right now.
It is now. I've now got it again. It's come again.
Okay, now give back the sharing rights. Yes.
Right. So I think we may be in business. So sharing content, and you should now.
Yeah, your whole screen is shared.
I don't see that at all. I think we're there now.
We can see the whole screen now.
Is that correct?
Yes. Yes, George. I think we are good to go now. Yes.
Right. Okay, so I'll start my talk. I'd like to start by thanking Professor Chaturvedi for his opening remarks, and also thanking Dr. Shyam Maskapali for his kind words of introduction. I'd also like to thank Shyam for the huge effort that he and his team have put into organizing this symposium. As some of you know, the original plan was to have an in-person workshop at Mandy, at IT Mandy last year, but the COVID-related travel restrictions made this impracticable. While we can't replicate the planned practical sessions in an online format, the video conferencing platform does allow us to engage with a wider audience. I'd also like to acknowledge the support from the UK-India Education and Research Initiative, and also from the Scheme for Promotion of Academic and Research Collaboration, for their support in running the symposium. And of course, it's great to be in IIT Mandi, albeit only virtually. So it's called FluxMAPS 2021, Fluxomics of Microbe and Plant Systems. So it has its focus on the metabolic networks that are found in all organisms. Metabolic networks supply the precursors, energy, and reducing power required for the synthesis and turnover of cellular components. The associated flows of material, the metabolic fluxes, are crucial in determining the performance and productivity of cells and organisms. For example, in an agricultural context, the production of harvestable end products of plant metabolism is entirely dependent on the flux phenotype of the plant. While in biotechnology, the exploitation of microorganisms and plants hinges on an ability to reconfigure the metabolic network to favor a particular flux distribution that leads to the preferential synthesis of desired products. Thus, the fluxes supported by a metabolic network play a pivotal role in determining both phenotype and productivity. So the title of my talk is Metabolic Flux Phenotypes, Predictions and Measurements. And my task in this opening lecture is to set the scene by providing an overview of the methods available for measuring and producing these network fluxes. It will fall to later speakers to explore these topics in greater detail, and where there is overlap between the talks, either today or tomorrow, the intention is that this will reinforce the message that network flux analysis can contribute greatly to our understanding of plant and microbial metabolism. Anyway, that's all by way of introduction, and as my next slide emphasizes, what you're going to get from me is an overview of work in this area, and it's going to draw on the work of many people over an extended period. And so I'd like to start by just acknowledging the people whose work I'm drawing on in this presentation. So when I'm talking about metabolic flux analysis, this is work I do in collaboration with Nick Kruger, who is also presenting in this symposium. And the people whose work I shall draw on, if only in passing, are Shyam Masakapalli, Maria Rossi, Ed Smith, and Tom Williams. 3 of those people are presenting in this meeting. And when I'm talking about flux balance analysis, then I do this work in collaboration with another close colleague, Lee Sweetlove. And here I must mention 4 other colleagues with whom I've done work, including 3 who are speaking today: Maurice Cheung, Sanu Shameer, and Nadine Töpfer. So where do we start? I'd like to start by just thinking a little bit about what flux is, and then move on, of course, to explaining how we measure it and predict it. So it's got 2 distinct meanings in biology. On the one hand, fluxes refer to the physical movement or translocation of ions and molecules into and out of cells, and also photons. That's a physical process, a movement, a translocation. On the other hand, flux refers to the flow of material between the inputs and outputs of a metabolic network. And that's a rather different type of flux. Because these fluxes, the metabolic fluxes, they describe the effect of a series of chemical changes. And there's no physical flow involved in necessarily in these chemical changes. And so, whereas you can imagine sensing the translocation of ions, for example, by measuring a current, metabolic flux sensing can only be indirect. And certainly in living organisms, which are not doing experiments on themselves, flux sensing, if it occurs, must indeed be some kind of indirect mechanism. Metabolite levels, or perhaps in some instances the enzymes themselves, might provide indirect readouts of metabolic flux. But whatever we understand by the term flux, we have to recognize that all biological activity is synonymous with non-zero metabolic fluxes. And so ultimately, when the fluxes stop, the organism has died. And it's because of this central importance in delivering the properties of an organism that it's important to focus on fluxes and to think of ways in which they can be measured or analyzed. And in tackling that problem, it's very important to take a system-wide view and to adopt the idea that what one's doing is network flux analysis, actually looking at multiple fluxes throughout the network and avoiding focusing on individual steps. And the aim is always in flux analysis, ultimately to come up with a map of flux, a distribution of the fluxes across part of a network or even a whole network. And just to emphasize the importance of network thinking, what you see here are some schematic flux maps based around the TCA cycle, the citric acid cycle. And these flux maps, what they show is in blue, blue arrows indicate fluxes that are significant. Things that are grayed out are insignificant. So what you see here are three more or less random flux distributions based on the TCA cycle. Maybe the one to focus on is the bottom right, where you see the experimentally deduced flux map for developing embryos of Brasilia. This is work from the Shakerhill and Schrender labs in the US. And what you see there, if you look at the TCA cycle, a very interesting flux distribution with fluxes going in opposite directions. There isn't a complete cyclic flux there, even though from the textbooks you would generally assume there would be a dominant cyclic flux through that cycle. So the point of showing you this slide is just to emphasize that the objective is to produce flux maps. After all, the conference is called FluxMAPS 2021. And flux maps are representations of the distribution of metabolic flux across a network. So as we get into this, it's important, as many of you will realize, to note that we can't necessarily think simply in terms of what the metabolite levels are doing if we're to understand fluxes. We will ultimately have to find ways of deducing or predicting those fluxes. But let's just explore the relationship between metabolites and fluxes just briefly. So what you see here is a metabolite with an unknown concentration X. And if that system that contains the metabolite is in a steady state, then X could arise, the concentration X could arise from a low flux in and a low flux out of that pool, or a high flux in and a high flux out from that pool. And what we're ultimately interested in when we're trying to understand metabolism is the fluxes. And so if we just pile in and measure metabolites, we don't immediately get any indication as to what the fluxes might be. Now, of course, we might not be in a steady state, and we might be in a situation where the metabolite concentration is increasing. And again, if X is increasing here, we don't know exactly what the influx and outflux from the pool is. 15 in, 10 out, 10 in, 5 out, 20 in, 15 out, 6 in, 1 out. They all would lead to the same rate of change in the metabolite X. And as a simple illustration of this lack of relationship between metabolite levels and fluxes, here are some measured fluxes, a fragment of the TCA cycle, and the size of the arrows indicates the magnitude of the flux. And as this flux through citrate increases, as you see here, it turns out that the actual concentration of citrate decreases. Now, that's a very difficult thing to reconcile simply in terms of the measurement of the citrate concentration. If you've measured citrate, you would not have been able to deduce the difference in the fluxes in this system. So the relationship between metabolite levels and fluxes is complex. And you can explore that if you wish, and as Nick Kruger and I did, in a little model like this. And here we've got a little network, and we've got a flux in here, J1, and it bifurcates at metabolite A and goes around two routes to product Y. And there are two other fluxes characteristic of the system, the flux round this way and the flux round that way. These are the net fluxes. And if you explore this computationally, what you find is that you get a very complex relationship between fluxes and metabolite levels. So to take one example, if you look here, what's happening here is that enzyme 3, that's this one here, its activity is being halved. And when you halve the activity of enzyme 3, what you find is changes in the fluxes through J1, J2, and J3. That's indicated by these bars, but really quite dramatic change between for J2 and J3. J2 is increased by about 70%. J3 has decreased by about 50%. And yet there's virtually no change in the metabolite levels. So even though there are large changes in flux, there are only very small changes in concentration of the metabolites. And if we do the converse and double the activity of the enzyme 3 in this network, again, we get very large changes in flux. The flux here through the J2 has dropped to a very low value, and the flux through J3 is increased in this case, but again, only modest changes in the metabolite levels. So all of this tells us that we can't focus on metabolites when we want to think about fluxes. We have to find ways of measuring those fluxes. So moving on, the focus has to be on building networks and measuring those fluxes. And these couple of slides are very straightforward. They just suggest that you might start with a genome annotation and produce some sort of network that looked like that. You might then refine that by prior knowledge and produce a network that looks like this. You don't have to start with a genome annotation. You might just start with your own knowledge of what you think the network should look like and start at this point. But you get some sort of network like this, and then you think hard about the thermodynamics, which really means deciding, well, some steps are irreversible effectively and some steps are reversible. And then, having got your network description, you can play with it. You can either do labeling experiments, and that is what will happen when I'm talking about metabolic flux analysis, or you can do pure modeling experiments, perhaps kinetic modeling or flux balance analysis, whatever it might be. And what will happen as a result of those activities is that you will generate a flux map, where now the arrows indicate the magnitude of the fluxes between nodes in the network. So that is basically how one sets about looking at fluxes. You've really got to start by constructing the relevant network. So where does that take us to? Well, we've got a network here. And the idea here is that this network underpins the growth and development of an organism, a rather fanciful plant shown here. And basically, the idea is metabolic inputs into the network or into the organism lead to a flux distribution in here, which we want to interrogate, that produces biosynthetic outputs that contribute to the development of the organism. So there's a circle around here of inputs and outputs. And lying in the middle of it, we've got this metabolic network. And the question is, how can we actually extract from the network the fluxes that are supporting whatever the organism is doing? And it turns out that there are really three approaches, and they're summarized at the bottom of the slide. On the one hand, one can take an experimental approach and introduce labels into a network. And these labels come in as inputs and they spread through the network, and you analyze the distribution of the redistribution of that label, either in real time or in a steady state. And that leads, in the process of metabolic flux analysis, to empirical flux maps. On the other hand, you might decide to do kinetic modeling, construct a network model, populate it with the kinetic parameters of the enzymes, and that will then lead you to a mechanistic flux map. Another possibility, which will be largely the topic for today, is to construct a network and then apply constraints to it and set it tasks, set it objective functions, and see how it can deliver those tasks. Well, I'm going to be talking about all three of these things and only briefly about kinetic modeling. So I'm just going to have a couple of slides on kinetic modeling before going on to the main topics, which are flux balance analysis and metabolic flux analysis. I would emphasize that these three methods or approaches, they're all valid, but each method depends on defining a network, and then the data requirements for the subsequent analysis will vary considerably. So these are three distinct approaches, all based on defining the network, and all with the intention of leading to these metabolic phenotypes that are going to provide insights into the structure of the network and their function and its function. So two brief slides on kinetic modeling. This brief because this is not the topic of the symposium, but just to emphasize that kinetic models require knowledge of both network topology, well, all these flux methods require that, and the kinetic properties of the pathway enzymes. And this rather abstract schematic, which comes from a review cited at the bottom of the slide, gives the impression that you have a network. This is a very simple one, a linear network where a substrate S is converted to a product P. It requires also a second substrate F, and something happens here. But the idea is there's a little network here, and you can balance this network. At any point A, you can say the rate of change of A, that's what this means, is equal to the flow into A minus the flow out of A. So these flows are represented as U and V here. And those rates, U and V, turn out to be functions of various concentrations of metabolites and intermediates in the system. So basically, this is just saying that for every step, this one is catalyzing that flux U, or this one catalyzing that flux V, there will be an enzyme. That enzyme will have particular properties. You know, it might be the classical Michaelis-Menten kinetics, or it might be something much more complicated. If you have a knowledge of the enzymes in your network, and you know their kinetic parameters, then you can make predictions about fluxes under any set of conditions you like. The difficulty, of course, is that you may not know all of the kinetic parameters for those enzymes, and this does put quite a limitation on kinetic modeling. But I would just emphasize that it is useful, even though I'm not going to be talking about it in this presentation. And just to emphasize that, I just highlight a couple of kinetic modeling studies from some years ago by groups that work in this field. And the point I want to make in highlighting these two studies is that kinetic modeling of plant metabolism led to biologically important discoveries about the systems that they were looking at. Of course, that's the whole purpose of flux analysis. We want, at the end of it, to learn something new about the way the metabolic network functions, the way the plant or the microbe functions. So in this first example, as it summarizes here, the kinetic model of sucrose accumulation highlighted a futile cycle that recycled more than 20% of the sucrose in the sugarcane internode. And that was a really interesting discovery for people interested in maximizing the production of sucrose in sugarcane. And as a second example, taken now from secondary metabolism, an analysis of the oil composition in peppermint, which is of interest to manufacturers of various products. What was discovered here was that there was a discrepancy between model simulations and the experimental measurements, and it led to a refinement of that kinetic model because they discovered a new way of regulating the production or the composition of the monoterpenoids that are formed in peppermint oil. I mention these two examples, which may or may not be of interest to anyone listening to this talk, simply to emphasize that you can take these kinetic models and learn important information about the way that the network is functioning and the plant is performing. In this case, we're looking at productivity. How much sucrose is the sugarcane producing? What is the composition of the peppermint oil? So I now want to move on to the first of the two main topics of the presentation, and that's metabolic flux analysis. So I've gone back to this schematic with the extraordinary organism at the top and the three approaches at the bottom, and now I'm going to be focusing on metabolic flux analysis. In broad outline, measurements of metabolic fluxes generally require the analysis of isotopic labeling experiments. And you've really got two options, I think. On the one hand, you can introduce the label into your network and follow it as it gradually disperses around the network. You can follow the dynamics of labeling. On the other hand, and there may be a good reason for not doing that, but we'll discuss that later, you might just introduce the label and allow it to redistribute around the network. And at a certain point, when you feel it's at steady state, then at that point, do the analysis. That would be called steady state metabolic flux analysis. And the principle is outlined here from a review that I wrote many years ago with Yair Shaka-Hill. And what you can see is here's a labeled glucose molecule. It has 6 carbon atoms. We're introducing 13 carbon, a stable isotope of carbon. That label is indicated by the black dot. It gets into a network and goes to glucose internal, which becomes glucose 6-phosphate. The glucose 6-phosphate might go to starch. That would take the label into starch. But also in this particular very simple metabolic network, the glucose 6-phosphate can move down to the level of triose phosphates, at which point the position of the label alters when you resynthesize the hexose phosphate. And that allows label in this product starch to end up both in C1, as it were, and C6. And the critical point in this steady state analysis is the third bullet point. The labeling of a carbon atom depends on the relative size of the fluxes that produce the intermediate. And so you end up in this simple system able to do a simple analysis like this, which shows indeed that the ratio of C1 to C6 does indeed depend on a ratio of fluxes. So this is the principle of steady state 13C metabolic flux analysis. You introduce a label into the system, you allow it to redistribute to a point where it is not changing anymore, and you then analyze that distribution and you are able to deduce fluxes. So this is a very well-established method, steady state metabolic flux analysis, often the preferred method for analyzing large-scale networks. And just in broad outline, the bullet points in blue indicate what you might do in a typical experiment. So you obviously have to introduce label into the system. And in many experiments, one might be using isotopically labeled glucose, which might be singly labeled or multiply labeled. you obviously have to verify that the system satisfies the conditions for the analysis, which is that it's an isotopic and metabolic steady state. And then you need a mathematical model that will describe that steady state and a method for extracting the set of fluxes from the data. And typically you do this by fitting the data numerically to an overdetermined model. And as it says in the summary at the bottom, this is really the method of choice for generating flux maps of the central pathways of, for example, heterotrophic and mixotrophic plant metabolism, but also in microbial systems. There's a great deal of work on both plant and microbial systems using steady-state metabolic flux analysis. And of course, we're going to hear lots more about this tomorrow in the second session. I would, though, want to just illustrate the application of this, just to give a flavor of what can be done with steady-state metabolic flux analysis. And in doing that, I'm going to be drawing on the work of Shyam Masakapali, just 3 slides, I think, to emphasize what's possible. So what you have here is the result of a lot of work that Shyam did many years ago, looking at the impact of nitrate and ammonium supply on heterotrophic metabolism in an Arabidopsis cell culture. And you don't need to look at the details, you just need to realize that I have 2 flux maps on the screen. One is labeled in this corner here as MS, which stands for the growth medium, and then there's another indication there. But the difference between these 2 growth media is that in moving from the left to the right here, we omitted ammonium from the growth medium. And okay, we got flux maps with and without ammonium in the growth medium. And the thing we had to do, Shyam had to do, was to look closely at the outputs of his analysis and realize that we were able to detect very specific changes in particular fluxes through the network. This is visually represented in the flux maps, but of course, ultimately, this is a truly quantitative method, and the output of the analysis are numbers that correspond to each of these fluxes. And these numbers, of course, have error bars on them that allow us to decide that there were statistically significant changes in fluxes as you move ammonium out of the growth medium, as you took the ammonium from the growth medium. Well, that's all very interesting and exciting if you're a flux analysis person, but what does it mean biologically? Because this is the reason you do this work. And Shyam was able to draw some very interesting conclusions, and there are 3 of them on this slide. The first was that flux through the oxidative pentose phosphate pathway varied independently of the demand for reductant for biosynthesis. Now, this is a fragment of the network, the oxidative pentose phosphate pathway, and it is involved in production of reductant for biosynthesis. But what he discovered was that flux through that pathway varied independently of the demand for reductant for biosynthesis. Well, you know, again, if you're interested in metabolism, that's a rather interesting conclusion, but you can extend that and realize that it highlights the complexity of the metabolic response and the difficulty of making intuitive deductions about the contribution of specific pathways. The whole point of going to these network analyses is to end up with a more correct picture of how metabolism supports growth and development of organisms. And it's a non-intuitive process. That's why we have to do flux analysis. He also discovered from the flux maps that non-plastidic processes made a significant and variable contribution to the provision of reducing power for the plastid. A very interesting conclusion if you're interested in re-engineering the reductant supply. And he was able to gain evidence for the futile cycling model of ammonium toxicity in this cell culture, bearing in mind that ammonium is not the preferred nitrogen source for Arabidopsis. This is great, and it was a very nice piece of work that Shyam did. But there are limitations of steady-state metabolic flux analysis, and I highlight 3 on this slide. The first is it's inapplicable to photoautotrophic metabolism. the problem here is that photoautotrophic metabolism depends on the assimilation of carbon dioxide. So the only sensible way to introduce label into your system is as 13CO2. But by the time the system has reached a steady state, because all the carbon is ultimately derived from CO2 in this autotrophic system, the 13C is everywhere and uniformly everywhere. And so there is no useful information on the fluxes that allowed you to reach that steady state. And to do this, we have to turn to a process called instationary metabolic flux analysis, where we relax the requirement for an isotopic steady state. I'll say something briefly about that in a moment. Another problem with the steady state MFA is and indeed with MFA more generally, is the confounding effects of cellular heterogeneity. In silico analysis demonstrates that labeling patterns averaged across cell types with different metabolic phenotypes do not reflect the aggregate metabolic activity. I'll illustrate that briefly shortly. And then finally, as you dive into your FluxMAPS, one of the things that you try to analyze are the coenzyme fluxes, fluxes involving NAD, NADH, and NADP and NADPH. You can only do that indirectly with carbon-13 MFA. There are possibilities using deuterium labeling, but then, as things stand, that method doesn't actually work in Arabidopsis cell culture because of the high rate of flavin enzyme catalyzed water exchange. It does work in mammalian cells. I'm not aware it's being tried yet in microbial systems. So I want to say just a little bit, 2 slides about photoautotropic metabolism to indicate how instationary analysis opens the way to analyzing that metabolism in a way that steady state MFA can't. So one of the pioneers in this field is Jamie Young over in the States. And this is an illustration from one of his papers. And the idea is to indeed introduce label as 13CO2, but rather than measuring a steady state, to measure the isotope labeling dynamics. So to follow the movement of label into individual intermediates, and to do this with a time scale that is comparable to the rate at which label is penetrating the system. So this is a very demanding exercise, which you'll hear much more about tomorrow from Ed Smith. In a different context, not a photosynthetic one, but it does work. And one of the most spectacular investigations of this was done by Doug Allen in the States. And what he was able to do was to construct a flux map of the photosynthetic metabolism in an Arabidopsis leaf using this method of instationary flux analysis. And what it was able to do was to provide direct measurements of the photorespiratory flux under different light conditions. The thing I would highlight, the thing I find most exciting about this extraordinarily impressive piece of work, is the way they were able to measure the rate of carboxylation versus the rate of oxygenation for the Rubisco enzyme in the illuminated leaves at low light or acclimated at high light. And you see this ratio, 3.5 to 1, 2.3 to 1. Hitherto, it had been very, very difficult to get reliable measurements of what is actually a critical ratio in determining plant productivity. So here's another very good example of a flux analysis method leading investigators to great insights into, in this case, photosynthetic metabolism. The other thing I want to highlight is the confounding effects of cellular heterogeneity. I won't go through the theory that shows you that the statement is true there. I just want to highlight that there is a possible approach to get around this problem. Of course, roots, for example, are full of different cell types, and we can ask all sorts of very specific questions about those cell types. You know, do the demands for energy, reducing power and carbon skeletons in a root hair cell, there's one up there, differ significantly from those in a cortical cell, one of these things? does the production of root exudates by a developmentally abnormal hair cell require changes in the metabolic phenotype? I mean, ultimately, there are very fundamental questions at a cellular level that one might wish to ask about the functions of specialized tissues. And we can't easily do that because we don't have our cell type specific techniques for flux analysis. You're probably aware from the analytical literature that, of course, there are great strides in cell type specific and indeed single cell analysis these days. I mean, it's possible to measure transcripts in single cells, but measuring those transcripts is a long way removed from understanding the metabolic phenotype. And the metabolic phenotype ultimately is the thing that drives development of the tissue you're looking at. So what you want ultimately is to be able to analyze metabolic phenotypes, flux phenotypes at a cellular level. And a possible solution is to use a reporter protein, but as I will indicate on the next slide, such a solution has not yet been fully implemented. So the idea here is shown in the slide. I mean, this is a representation of an Arabidopsis root, and it's got a specific expression in the endodermis here of a 13-carbon labeled GFP. Well, no, this is just GFP. This is just green fluorescent protein. And the idea is that, of course, if this root has been, as it were, programmed through genetic modification to express the GFP in a particular cell type, then perhaps we could use that protein, which is a cell-specific protein now, as a reporter of metabolism in those cells. So the idea would be that you introduce labeling into this system, 13C label. That 13C label at steady state gets into all the structures of the plant root, including the GFP, which happens to be localized in a particular group of cells. So the idea is that you purify that GFP and you analyze the 13-carbon labeling of the constituent amino acids, and from that deduce the metabolic fluxes. And Maria Rossi looked hard at this, and indeed she was able to make a lot of progress to demonstrate that she could purify constitutively expressed GFP to a very high level of purity using a GFP binding nanobody. The labeling of the GFP matched the labeling of the total protein fraction, and that the expression of GFP didn't perturb metabolism. But she ran into a roadblock because the sensitivity of the mass spectrometry techniques emerges as the principal obstacle to implementing the strategy. Although you can purify the GFP to a high level and get through to levels of amino acids, it's very hard from the relatively small amount of GFP that you extract from the tissue to generate mass spectra that you can really interpret. So for the moment, that's for the future, but I'm sure mass spectrometry sensitivity will eventually allow this problem to be solved. In the remaining part of the talk, I want to touch on flux balance analysis and give you an overview of this. This is the third of the approaches, as I indicated earlier. This permits the prediction of flux distributions that are possible when you convert substrates to products under specified conditions. So the idea is you build a stoichiometric network model. I'll say a little bit more about that in a moment. And you say, I want this network to do something. And that you then work out how the network can do that thing. Now, of course, you have to constrain the system and maybe give it some inputs, and you have to decide what you want the network to do. But that is in essence what you do. You predict the flux distributions that permit the conversion of substrates to, say, products under specified conditions. And you do it using a stoichiometric model of the network. And these models define the relationship between the metabolites in a reaction network. So in this little fragment here, A and B together make D. D can also be derived from C. D can, as well as being produced in these ways, can produce E, and so on. And the idea is that what you do is you construct a stoichiometric network that just relates the metabolites and indeed the atoms within the metabolites according to the stoichiometries of these conversions. So as you see here, D goes to E. So if we look at reaction 2, reactions are specified by the columns. Reaction 2 here, we see that D, we've given a -1 here, goes to E, a +1. All we're doing is defining the relationships at this point between metabolites in this network. And we're reducing it to what's called a stoichiometric matrix. It seems a very abstract description of a metabolic network, but it's extraordinarily powerful. And one of the things you can do with it is flux balance analysis. So you build a metabolic model and you construct its stoichiometric matrix. And then what you're going to do is you're going to apply some constraints to that model and set the model a task, an objective function, and you end up with prediction of the fluxes. And in embarking on this, what you have to recognize, and this is very important, is that these solutions, these predicted fluxes, are not going to be unique. They could conceivably be unique in certain circumstances if you really close down the model, but generally, FBA solutions are not unique. And further constraints are added to reduce the solution space and the variability of the predicted fluxes. The point is, if you set up one of these stoichiometric models and apply a few constraints and ask the model to do something, there may be lots of ways of doing that. Moreover, when you solve this problem, the computational tools will only generate one solution immediately. They will produce the optimal solution to achieve what you want. And that will be typically one of many solutions to the problem. And there are ways of investigating it and interrogating it, which are essential for doing FBA. But nonetheless, the principle is clear. There are many solutions to a typical problem you might set the network, and the computational methods will initially just give you an optimal solution that is one of a family. So we're going to be interested ultimately in the size of the solution space and also the variability of the predicted fluxes. And so I won't be going into much detail on this in my overview, but I'm sure this will emerge in some of the later talks. So a next point to make is that this flux balance analysis is applicable to large scale, even genome scale networks. This is actually a small network because it's only got about 600 reactions in it. It's actually one that Sanu Shameer uses, and he'll be talking about later. It's, as it says, a charge and proton balance model of leaf primary metabolism. And it's got 600 reactions in and more than 500 metabolites. And of course, when you see networks presented like this, you realize just how big they are and how naive it is to assume that we can really understand how they function intuitively just by focusing on this little bit here because it looks interesting and thinking, oh yes, we understand that, and then we understand that. But there's crosstalk between all the parts of these networks, and that's what complicates our understanding of them. So how do you do flux balance analysis? Well, you have to think about constraints and objective functions. And constraints restrict the solution space defined by the stoichiometric matrix for achieving a particular objective. And there's a whole pile of constraints that you might use. These are some of the possibilities listed here. We might be interested in defining the inputs into the system, which might be mineral nutrients or photons. We might be interested in defining what we want the network to achieve in terms of an output flux. Constraints around enzyme availability. It is possible to put all sorts of weightings into your network according to, for example, transcript abundance of a particular cell type, if you choose. All these networks need to be mass conserved, but also they can be charge conserved. And thermodynamics comes in where you think about reaction reversibilities and whether, for example, some reactions are just not thermodynamically feasible and so should be excluded from the network. You will hear much more about constraints later today. I'm giving you the principle. We have to apply constraints, but we also have to set the model an objective function. We have to say, for example, we want you to convert inputs to outputs subject to, and then you have an objective function, which might be, for example, maximizing growth rate, or if you're in a leaf, maximizing phloem output. Or you might decide you're interested in maximizing ATP yield or minimizing substrate consumption. There's a lot of different objective functions, a lot of different constraints. And choosing these is part of the art, if you like, of doing effective flux balance analysis. So the selection of constraints is very important in determining the validity and usefulness of the predicted flux distribution. And I want to illustrate this with a piece of work that now Sanu Shameer did in Oxford. So this is to, I'd forgotten there's an important slide to emphasize that FBA and MFA are similar, but agreement is not guaranteed. And the reason for this is obviously spelled out in the bullet points, but on the one hand, carbon is being tracked in metabolic flux analysis, and it is giving us real values for the net and exchange fluxes in the system, but we're only tracking carbon flow. In contrast, what flux balance analysis is doing is predicting an optimal flux distribution, which may or may not correspond to reality. On the other hand, it's also redox balanced and energy balanced, and it's consistent with the defined objective function. These are two very different approaches, and they don't always agree. They can agree, and often you will find papers where they do agree, but I want to illustrate a situation where they didn't agree from a nice piece of work done in the Shaka Hill lab, where they were looking at the growth of an E. coli strain. And this plot here is a plot of growth rate against oxygen uptake rate. And measured values of oxygen uptake are shown here, and measured values of growth rate are shown here. And predicted oxygen consumption from MFA is shown here. But the flux balance analysis threw up the possibility that you could be anywhere in this colored range here, and that this would be the best place to be. So here's an example where an FBA prediction says you could be here, but what reality tells us is we're here. And what that led to was, of course, some biochemical interpretation and led to the conclusion that there was a restriction on oxidative phosphorylation in this system. So the comparison between FBA and MFA can be very informative. While one might set out doing these things in parallel and hoping that they agree with each other, the differences can also be very, very informative. So now moving on to a specific application of FBA to plants, and noting that there are all sorts of extra things that we need to think about, and they're shown in blue here, not least cell maintenance costs, but also all these other problems that you associate with plant cells, such as subcellular compartmentation and metabolism, and so on, things which will emerge in later talks. Nonetheless, despite all these complexities in the structure of plant cells, all sorts of modeling strategies have been developed, some of which are listed here, all of which I think will be illustrated in the course of lectures later today. But as I said a few minutes ago, what I want to do is just highlight a particular application that's been carried out by Sanu Shameer, where he was interested in a very important problem, energetic coupling between organelles in an illuminated leaf. And this was explored using DL-FBA, which I'll explain in a moment. But the key thing here is look at the complexity. There are all these processes which are allowing ATP and reducing power to be produced and moved about between compartments. And it's a central problem in leaf metabolism to understand the balance, the energetic coupling between these different compartments. And what you find is that the only way to do this with flux balance analysis is to go for a DL modeling framework. And this was a very important concept introduced by Maurice Cheung, where the idea is you simultaneously model what goes on in the light and what goes on in the dark. And this requires duplicating the model between different phases and having linker fluxes between them. This turned out to be a very important development because immediately it led to the possibility of getting realistic predictions about leaf metabolism. And I highlight just a couple here. The DL model, in contrast to a single light steady state model, actually makes correct predictions about the source of carbon skeletons for the synthesis of glutamate in the light. That was a very important prediction from flux balance analysis. This is borne out by experimental work. And it also, using a DL approach, allows you to predict the existence of non-cyclic flux modes in the TCA cycle, which again is how leaves actually function. And again, it only emerges when you set up this idea of a two-phase model. Well, Sanu took this approach and set up such a model and analyzed the leaf metabolism in much more detail with a view to focusing on the transport of energy between organelles. The blue here, I'm not going to go through, that tells you how he did it. And what emerged was a set of flux maps. I'm only going to highlight the statements in green at the bottom of these three slides. The first thing he observed was that using FBA, he could demonstrate that the chloroplast could meet the bulk of the cytosolic ATP demand in both source and growing leaves. Note that what we're interested in is the movement of ATP and reducing power between compartments. It might be produced in the chloroplast, or ATP might be produced in the mitochondrion. How, for example, do we meet cytosolic ATP demand? And he came up with that prediction. But it was a surprising prediction, and it's not actually compatible with known activities for some of these transporters. So the next thing was to constrain the model further, as it says here, with constraints on the export of chloroplast ATP. And what that did was to change these flux maps. We go from unconstrained export to constrained export. And what you've got is a different flux map. So what we've got now is that actually we require substantial ATP to produce in the mitochondrion under conditions of realistic export from the chloroplast. And you go further than that and you say, well, maybe we're not using all the light energy that might be available in the system, and you can investigate that. And you find that if you do that, then at every point you're going to be using mitochondrial ATP to make ATP for the whole leaf. And that leads to the conclusion that in a leaf, mitochondrial ATP synthesis is more energy use efficient and a better way to meet the cytosolic ATP demand. So can you see we're getting deeper and deeper into understanding how the leaf works? until we end up with some conclusion such as this shown at the bottom. But what I'm emphasizing here is that in order to explore the behavior of the model, of the network, you have to think very hard about the constraints you put on the system and then to examine what happens as those constraints change. Of course, this nice piece of work of Shameer's at Sanu's produced some very important biochemical conclusions, which are summarized on this slide. They will only be of interest to people listening to this talk who are interested in leaf metabolism. But the big take-home message was that the chloroplast malate valve and the triose phosphate 3-phosphoglycerate shuttle are predicted, remember these are predictions, have an important metabolic role beyond their contribution to the avoidance of photooxidative stress. Now that conclusion is of great interest, I think, to people who work on leaves. It may not be of great interest to those listening to this talk, or to only a subset of them. So I come to the concluding slide, the summary. There's a series of points here to emphasize. MFA and FBA provide complementary methods for analyzing the flux distribution in compartmented plant metabolic networks. I haven't said much about microbes. We'll hear more about that later. Microbes also are easily studied in this way. Of course, there have been lots of technical developments which have expanded the range of these techniques, and they lead to biological insights into network function. And that in turn helps us to understand how we can manipulate networks and make them more productive for our own purposes. Of course, the MFA and FBA differ in these two in bullet points shown here. And ultimately, this constraints-based modeling approach of FBA is a more powerful technique for exploring the capacity of a network, provided the model is not over-constrained with experimental measurements. That's the final take-home message. And when you go into flux balance analysis, you have to be careful not to constrain the model to the point where it tells you nothing at all other than what you knew at the beginning. So at that point, I'll end and thank you for your attention.
Yeah, thank you, George. That's for such a fascinating talk, especially wherein you covered metabolic flux phenotypes, mainly the value of predictions as well as measurements and their comparisons in terms of MFA versus FBA, and the whole journey across several years of work. Really fascinating. So we do have some questions from the participants, which I would like to put forth for your inputs or for the discussion. Any panel members can also add to that. One question is, does measurement sensitivities influence fluxes? Is one of the questions which we received. I'll take one by one. Maybe you can just give your views on that.
Well, measurement sensitivity is critically important. Certainly, if you don't have sufficient sensitivity in the definition of the labeling of the intermediates that you're analyzing, then you won't end up ultimately being able to deduce as much information about the fluxes that led to the labeling. You do need high sensitivity. Of course, if you go for mass spectrometry, then often it will give you sufficient sensitivity to define the labeling patterns, although I gave you an example where it didn't, with the inability so far to get sufficient amino acids out of labeled GFP. So yes, measurement sensitivity is important. People mostly these days use mass spectrometry, but NMR is also a very valuable way of getting positional labeling information. But ultimately, you always need high measurement sensitivity if you're to have confidence in your analysis of the labeling patterns.
Okay, thank you. I think that answers. Another question is, how can we differentiate between the metabolic fluxes when intracellular compartments are involved?
What you need to have there is information that relates to the behavior of the individual compartments. So, for example, in the case of the heterotrophic Arabidopsis cells, the starch in the system is produced within the plastid. And that means that if you analyze the starch or the glucose derived from the starch, then you get information about labeling that has occurred specifically in the plastid. So you need compartment-specific information if you're to probe subcellular compartmentation.
Yes. Yeah, so thanks for that. Now, the next question, I will also read actually who has been asking the question. So this is a question from Berkeley Walker. Most of these approaches appear to require a metabolic steady state assumption. Do you have any thoughts for approaches that investigate non-steady state situations? Are reaction kinetic models the only current option for these situations?
Well, I mentioned so-called instationary analysis only briefly, but there will be a talk tomorrow from Ed Smith describing the instationary approach to metabolic flux analysis. And the key point here is that is not a steady state approach. That's where you look at the dynamics of labeling, and that greatly extends the scope of flux analysis. But it comes at a cost because it is very demanding analytically. The number of applications of instationary metabolic flux analysis is relatively small.
Okay, thank you. I hope that answers Berkeley's question. And we do have one question from Mr. Yogesh Pant. He asks about, I would like to know more about various applications of deciphering FluxMAPS.
Could you repeat that? I didn't quite catch it all.
Yes, George. So the question is, I would like to know more about various applications of deciphering FluxMAPS.
Well, the first thing to say is that if you last the course and spend 2 days listening to all the lectures that are coming, you'll find a huge range of applications. I think probably we have to wait for the later talks rather than providing an answer to that question.
Yeah.
The next talks.
Yeah. So another interesting question from Mithil Gaikwad. Is there any method to study intercellular FBA? Intercellular FBA. Is there any method to study intercellular?
So intercellular, looking at interactions between cell types. Indeed there is, and there will be illustrations of that again, I'm sure, in the later talks. That's the idea. But certainly you can set up models which have more than one cell type. You can set up models that have more than one tissue type. There was going to be a lecture, I think, on that this morning, but I think it's probably not happening. Is that right, Cheyenne?
Yes.
The lecture from Tarit, is that had to be cancelled?
Yes, it is cancelled, but we'll take
yes,
on
okay. But certainly you can set up models which include multiple cell types, multiple tissue types, and conduct flux balance analysis on them with suitable constraints, of course, on those cell types. So yeah. There will be illustrations, I'm sure, during the course of the day.
Yes. And again, tomorrow we do have the challenges of studying interacting organisms as well. If there are more than one, like plant microbial interactions and so on. So maybe that would answer some of Mithil Gaikwad's queries as well. We do have one more question from Bertrand Gakier. Sorry if I'm spelling the names wrong. Sometimes we are looking for bottlenecks in pathways that are similar between different subcellular compartments. So do you think labels and NMR techniques could identify the more productive pathway without having any dynamic measurements?
That's a complicated question, which I don't fully understand yet. Could you just repeat it?
Yeah, I'm just repeating it. Do you think labels and NMR techniques could identify the more productive pathway without having any dynamic measurements?
The most productive pathway. So here one's envisaging a situation where there might be two pathways to some product, and one is more favored than the other.
Yes, I think that is what Bertrand want. He's looking for, say, for example, we are looking for bottlenecks in pathways which are similar between different subcellular compartments. Now,
well,
can just labels provide the information? So that is the question.
Well, what labeling will do is ultimately allow you, if you're fortunate, to measure the fluxes through pathways and therefore to quantify which pathway might be more important than another pathway. And so in that sense, you would learn something about the relative importance of pathways. And you might then say, well, the pathway which is not carrying so much of the flux of the product is perhaps some kind of bottleneck, but I'm not sure I would use that word. I'd be a bit cautious. And certainly learning about parallel pathways to a product is an important thing to be able to do in some circumstances. How easy it is would depend on what your labeling strategies were, and you might then be using either NMR or mass spec. Does that help?
I hope so. Yeah, and definitely you've given some indications of how that can be done. And here, maybe final two questions, George. While I encourage others to type their questions, we can take up later also. The final two questions, out of which first one is from Majia. For a whole plant which is having like multiple compartments, metabolic model, is it suitable to have the biomass reaction or objective function in the cytosolic compartment?
That's interesting. Different organelles could indeed be associated with different objective functions. Yes, you have to think hard about this. The more complicated your model, and I sort of gave a brief indication of that in talking about Sanu's work, how the nature of the constraints that were applied gradually became more constricting, if you like, and forced the behavior of the model towards a particular point. So yes, when you're thinking about biomass equations, you have to think about where the biomass is being produced. But in general, and this is something one would instinctively do, I think, in leaf models, for example, of course, the leaf is highly compartmented. Often the output flux that we're interested in is export to the phloem. So in a sense, that then becomes the production of the whole leaf cell. And so it becomes easier there. But yes, I mean, it's an important question to consider, what is happening in different compartments.
Yes, correct. Thanks for that. And here we have one final question, actually two final questions. Sorry about adding one more, is very interesting. So how can we metabolic flux for differentiating? It should be, how can we use metabolic flux for differentiating root exudates of plants in rhizosporic microbes?
I'm not sure I understand that question, Shyam. I think we should move to the final question given the time.
Yeah. The final one. Okay. Is there any paper or book which can teach us this kind of analysis?
Oh, well.
So I thought
there are lots of review articles that teach you this sort of work. Of course, you're going to learn a lot from the next 2 days listening to the talks. But the review literature is the place to go, I think. There are 1 or 2 books on metabolic flux analysis, but books are always harder to find than papers. But there's an extensive review literature. If you, I mean, I had 1 or 2 reviews on the list of papers that were included in my talk. And no doubt other speakers will also have references on some of their slides. So it is a case of diving into the literature.
Yeah, I agree with that. And the question was arose by PK Rai. So we will definitely provide you some inputs on that in the coming lectures. And with that, I thank George for such an exciting talk, as well as for the keynote in particular, which now has set up the platform for the symposium to go ahead. Thank you very much, George.
Thank you, Shyam. I shall return at 10:30 exactly.
Yeah, so right now you
Can you
can
stop sharing my screen?
Yeah, you can stop, or
Apparently not.
just let me just check participants. Yeah, Chandrakant, can you please give me the sharing privilege? Yeah. Okay, so yes, sorry. Okay. Yeah, if we go. Okay, this is for everybody actually, all the panelists and other. Now, right now we'll have a break of about 10 minutes, and then we'll again catch up at 4 p.m. That is Indian Standard Time, or UK time of 10:30 a.m. Okay, you can all either log off and log in again. We will stay online. Stop the sharing. Maybe I can go to the, yeah. Yeah, so welcome back everyone. Let us start with the next session. Chandrakant, may I request you to flash the next session? Just go to the next slide. Yeah. So we'll be starting with the next session, FBA in plant systems and case studies. So George, request you to chair the session and introduce Nadine.
Okay, thank you. Are you going to show the picture of Nadine and?
Yeah, Chandrakant, yes.
Right, okay. So it's my pleasure to introduce Nadine Töpfer, who's going to give this next lecture. As you can see, Nadine received her PhD from the Max Planck Institute of Molecular Plant Physiology, and she did postdoctoral work in Israel and also at the University of Oxford. Importantly, she's now an independent research group leader at the Leibniz Institute of Plant Genetics and Crop Plant Research in Germany. And she's going to give us the next talk, which will be a description of flux balance analysis. Over to you, Nadine.
I can't share my screen at the moment.
Now you will be able to share your screen.
Okay, just give me a second to set myself up.
Excellent.
Good. Okay. Thanks, George. Thanks a lot. I'm actually very excited to be talking at this meeting today, and I've been really looking forward to it. And as you said, I was doing a postdoc at the Department of Plant Sciences in Oxford, and in preparation for this meeting today, I was thinking back of these times, and it really felt like I left only a month ago, but it's been a year and a half already. And a lot of things have happened in the meantime. Yeah, so I've started my own group now, and we're also continuing our work on modeling plant metabolic systems. So today I'll be giving an introductory talk to flux balance analysis and its application to plant systems. Okay. So the clicking isn't quite working. Okay, here we go. So to give you an outline of this presentation, I want to start with a motivation as to why it's important to study metabolism and metabolic fluxes, then give you some theoretical background on, I will talk about kinetic modeling and also about stoichiometric modeling and actually make the connection between those two. Then I want to briefly talk about some applications of flux balance analysis to plant systems, basically giving you an outline of what you're going to hear in these various talks today. And then talk a little bit about my own work that I did in Oxford, where we were studying water-saving flux modes in a leaf metabolic model. So why is it important to study metabolism and metabolic fluxes? So if you look at this flow chart here from the genotype to the phenotype, so here the genotype is represented by the DNA, you can see that any changes that occur on the level of the DNA, RNA, protein level can directly or indirectly affect metabolism or rather the metabolic network. And it's important to talk about the metabolic network, and I will explain this in a second, and thus eventually will affect the phenotype. So any changes that occur on the level of metabolism have or can have a direct impact on health and growth of a plant. So why is it now important to consider metabolism on the network level? So the thing is that metabolite levels and metabolic fluxes are inseparably connected. So changes that occur on the level of the metabolites can affect the metabolic fluxes, and in return, changes of metabolic fluxes can affect the level of metabolites. So really any changes that occur on individual metabolite levels or on individual fluxes can have an impact on the whole metabolic network. So this is why it's really important to look at metabolism as a network. Now, when it comes to analyzing metabolic fluxes, we basically have two main approaches. So we can either measure metabolic fluxes by means of metabolic flux analyses. So this typically requires the incorporation of labeled carbon, and you will hear a lot about this tomorrow. On the other hand, we can estimate metabolic fluxes by means of either kinetic or stoichiometric modeling. So if we're looking at kinetic modeling approaches, we need to have information about the kinetics of the involved enzymes, and we also need to have knowledge about initial conditions. Whereas if we're looking at stoichiometric modeling approaches, or FBA in particular, typically we need to have knowledge about uptake rates, and we need to have knowledge about an optimality assumption. So as you've already heard, today we'll be mostly talking about stoichiometric modeling and about flux balance analysis, or FBA in short, in general, but I also want to briefly talk about kinetic modeling. So let's dive into the theoretical background. And what I'm showing you here is a small toy model. So this could be, for example, a leaf cell, a mesophyll cell, which has three metabolites, metabolite A, B, and C. And these three metabolites are connected via reactions. So we have reaction 1, 2, 3, 4, 5, and these reactions can carry a certain flux, which is denoted by the V. So if we now want to look at the dynamics of the system, so how the levels of the metabolites and how the fluxes through the reaction change over time, then kinetic modeling would be our approach of choice. And if we want to do kinetic modeling, we need to write down a system of equations. So what does it mean? So if we want to look at the dynamics of our system, we actually want to look at the rate of change of the concentration of a given metabolite over time. So this is denoted here, dA over dt. So that's the rate of change of metabolite A over time. And this is basically defined by the sum of all incoming fluxes minus the sum of all outgoing fluxes. So in this particular case, this would be V1, because V1 is an incoming flux, it's producing A. And this would be minus V2 and minus V3, because those two fluxes are actually consuming metabolite A. Now we can write down the same equation for the rate of change of metabolite B. So metabolite B is produced through reaction 2, and it's being consumed in reaction 3 and 4. And the same for metabolite C, which is being produced through reaction 3 and consumed in reaction 5. Now, if we only look at the system of equation as it is here, we can't really make any statements about the dynamics of the system or what is really going on, because we need to have information about the kinetics of the individual reactions. So if we assumed the most simple form of kinetics, there would be mass action kinetics. And if we were to write this down in form of mass action kinetics, then V1 would be equal to K1, so this would simply be a constant influx. The flux through reaction V2 would be K2, so another constant, times the concentration of metabolite A. If we look at V3, this would be another constant, K3, times the concentration of metabolite A times the concentration of metabolite B, because both metabolites are required for this reaction to proceed. And in a similar way, we can also do this for V4 and 5. However, if we're looking at a more realistic scenario, if we're looking at Michaelis-Menten kinetics, we would have to have knowledge about the Vmax values of the involved reactions and also of the Km values. So as you can already see, in order to perform kinetic modeling, we need to have extensive knowledge about the kinetics of the underlying reactions. So this can come either from experiments or we need to estimate these parameters. However, let's assume for this small toy network, we do have knowledge about all the kinetic parameters and we have knowledge about initial concentrations. Then we can simply solve our system of equations by using an ODE solver. And what we obtain is the dynamics of the network. So if we're looking at here, for example, the concentration of the 3 metabolites, which are color-coded over time, this could be a typical profile that we obtain by using our kinetic modeling approach. And as you can see, as time progresses, the system is moving towards a steady state. So that means the concentrations of the involved metabolites do not change anymore. And this will be important in a second. So to quickly summarize, in order to do kinetic modeling, we need to have knowledge about the kinetic parameters of the involved reactions, and we need to have knowledge about the initial conditions. So, however, in reality, this might not always be the case. So, especially when we're not only looking at 3 metabolites, but if we're looking at a large-scale metabolic network, which includes hundreds of metabolites and hundreds of reactions, so then kinetic modeling might not be the suited approach anymore, and we can apply stoichiometric modeling. So stoichiometric modeling has the advantage that it doesn't need kinetic information for the involved enzymes. However, stoichiometric modeling can only model steady-state behavior. And I will show you now how kinetic modeling and stoichiometric modeling is actually connected. So again, if we look at our system of equations, I'm just rewriting the system here a little bit by putting in the stoichiometric coefficients of the involved reaction. So instead of V1, we have 1 times V1, minus 1 times V2, minus 1 times V3, and we put in a 0 because metabolite A is not involved in reaction 4, nor is it involved in reaction 5. And we can do the same thing for the other metabolites. And if we look at this now, we can see that this actually corresponds to our stoichiometric matrix. So this basically is a matrix containing all the reaction stoichiometries of our system under consideration. And if we look at this row, we see that this is our flux vector. So in each of these rows, we have the flux vector. So what we can basically do now is to simply separate these two and rewrite the system of equations as the stoichiometric matrix times the flux vector. And if we do some matrix multiplication, we get back to our initial system of equations. So as I said, in order to perform stoichiometric modeling, we need to assume that we're in a steady state. Steady state means that the rate of change of the metabolites involved in our system is constant. So we don't have any changes. Thus, we can set our system we can set this equal to 0. So this is basically the short form of what is written here, S times V equals 0. So this is our steady state assumption. So again, now in a similar way as with what I showed you earlier when I talked about the kinetic modeling approach, if we don't have knowledge about the enzyme kinetics of our involved reactions, we can't really tell which reactions are active. So we could have fluxes going through reaction 1, 2, and 4, or we could have fluxes only going through reaction 1, 2, 3, and 5. So we don't know this. So In order to make predictions about our system under consideration, we need to apply certain assumptions. We need to apply an optimality assumption. And this could be, for example, so if this was a growing leaf cell, this could be the production of biomass, which we would then call the biomass function. So the biomass function is nothing else but a defined ratio of biomass precursors that are required in order for the leaf to grow, so for the leaf to produce new cells. So we can then plug this into our optimization problem. So we would state maximize biomass such that S times V equals 0. And then we also need to, or we can apply some other constraints. And as George pointed out earlier, this could be a whole range of constraints. So for example, known uptake rates, information about the thermodynamics of the involved reactions, or also known excretion rates or known biomass production. So this is basically the basic FBA or flux balance analysis formulation. And if we now go back to the scheme that I showed you in the beginning, we can't simulate the dynamics of the network, but we can actually look at what is happening in our metabolic network in a steady state. So to summarize also at this point, what is required for stoichiometric modeling is, apart from the stoichiometry of the involved reactions, is a smartly chosen objective function and typically knowledge about exchange fluxes. So in the third part, I want to now come to talk about some applications of flux balance analysis to plant systems. And I want to start this by showing you an overview slide. An overview slide of different areas or different angles, different topics, which can be worked on, and really where metabolic modeling can help us in tackling important biological questions. So today we will hear about plant environment interactions, so with a particular focus on the leaf. So this will be part of my own presentation where I'll be talking about water-saving flux modes in a leaf metabolic model. And then later, Maurice will zoom in and have a closer look at the leaf, and he will talk about guard cell metabolism. Apart from the leaf, we can also look at other organs. So for example, we can look at seed metabolism or fruit metabolism. So Sanu will enlighten us later, and I think he'll be talking about modeling tomato metabolism. So during tomato growth by simulating osmotic cell expansion. We will also have a talk today related to plant microbe interaction. So George will talk about modeling the metabolism of nitrogen-fixing legume nodules. And we will also focus, so we will start integrating various organ-specific models into a whole plant modeling framework. So Tarit will talk about source-sink interactions in a whole plant model, and Thomas's talk will be sitting somewhere in between seed metabolism and whole plant metabolism, I believe, because he's talking about flux balance analysis of seed reserve mobilization. And last but not least, we will also have a more technical and method-driven talk today. So Chandrakant will be talking about efficient system-wide pathway comparison in multiple organisms. So having this said, I would like to come to the last part of this presentation. And I want to talk about the work that I mainly did during my time in Oxford. And I will tell you a bit about water-saving flux modes in a leaf metabolic model. Now, why is it important to understand leaf metabolism, and especially leaf metabolism with respect to water use efficiency? Well, the thing is, the world is becoming increasingly arid, and currently we're using roughly 70% of our fresh water in agriculture. So here you can see a stock image of a field, but this is basically what it also looked like in Germany in the past 2 or 3 summers. So water use efficiency really is important when it comes to understanding, or also when it comes to engineering leaf metabolism. Now, it's important before we continue with this talk to really understand that plants lose water through transpiration. So it's really not the water that is involved in metabolic processes. This is really insignificant to the amount of water that is lost through transpiration. And this happens when the plant opens the stomata on the leaf surface to allow for gas exchange, so to take up CO2. During this process, when the stomata are open, plants inevitably lose water through the transpirational stream. However, nature has already made some great inventions to circumvent or to kind of counteract these problems, and we have different types of photosynthesis. So there is C3 photosynthesis. So C3 is what most plants are doing, and in C3 photosynthesis we have a direct fixation of CO2 that enters the leaf. So it directly goes to rubisco and is fixed in the Calvin-Benson cycle. But then there are 2 alternatives. There's C4 and CAM photosynthesis, and these alternatives are particularly suited to for plants growing under hot and dry conditions. So in C4 photosynthesis, the initial step of carbon fixation and the refixation step by rubisco is spatially separated. So C4 plants really have a carbon concentration mechanism which increases the concentration of CO2 around rubisco, and this then suppresses photorespiration. Photorespiration produces byproducts that need to be expensively recycled. And this dual activity of rubisco that is causing these byproducts to be synthesized is really temperature dependent. So C4 photosynthesis is really efficient in high temperatures because it's suppressing photorespiration. Then there is another type of photosynthesis, and this is CAM photosynthesis, where these 2 processes, the initial fixation and the actual refixation by rubisco, is temporally separated. And this is what is going to be the center of the remaining couple of minutes in this talk. So here you can see a few examples of typical CAM plants. And if we look at CAM photosynthesis, or really crassulacean acid metabolism, so here I'm showing you an overview of the most generic scheme. So what happens in CAM photosynthesis is at nighttime, when the temperatures are low and relative humidity is high, the plants open the stomata, CO2 can enter the leaf, can enter the system, and it's then being fixed by PEP carboxylase to form malate or another carboxylic acid, which is then temporarily stored in the vacuole. Then during the day, when the temperatures rise and relative humidity becomes lower, the plant can close the stomata and the carboxylic acid is released from the vacuole. It releases CO2, and this CO2 can then enter the Calvin-Benson cycle and be used by rubisco to eventually synthesize starch, which is then stored in the plastid. And now to complete the cycle, at nighttime, starch is being degraded in order to sustain metabolism, but also to regenerate the precursor PEP, which can then serve as a precursor for the next round of carbon fixation. Now this CAM cycle is, well, it is known to be a water-saving mechanism, but it's also thought to be energetically more expensive than classic C3 photosynthesis. And another thing that I want to point out is that although there are morphological differences between chem plants and C3 plants, so chem plants typically show succulence, basically any C3 metabolic network or any C3 plant already has the necessary set of enzymes to run this chem cycle. And there are actually plants that can shift from C3 to chem photosynthesis under inductive conditions. And such an inductive condition could be, for example, salt stress. So when I started this project, our initial questions really were, so given a C3 metabolic network, are there any alternatives to the classic chem cycle? And if yes, what could such a water-saving system look like? And what are the limitations to it? So if you followed my presentation and what I was saying in the beginning, you'll actually notice that we do have a bit of a problem here, because I'm talking about modeling chem photosynthesis, and this requires including a temporal aspect. Although earlier I was talking about that FBA can only simulate steady state metabolism. So this is a problem that we needed to solve. So we really had to come to a description in order to represent the ideal leaf metabolism, so to really be able to show the dynamics over the course of the day. And then the other problem is transpiration, because I told you earlier that transpiration is really the main driving factor that's causing the plant to lose water. But then also I told you that FBA is suited to simulate metabolic models. So we somehow have to come to a representation of the water loss through transpiration. So this was the second challenge, to come to a feasible representation of the gas-water exchange through the stomata, and then to couple it to the metabolic model. And then once this has been done, we can actually study the trade-off between leaf productivity and water saving. So this was kind of an outline of what you'll be hearing in the next couple of minutes. So let's really look into the modeling part. So we started this project with a core model of plant metabolism. When I'm talking about core model, I mean that we're looking at a generic model of primary metabolism, which accounts for roughly 600 reactions and 500 metabolites. Now, in order to come to a description of the deal behavior, so to come to a description of the dynamics over the course of the day, I took 24 copies of the same model, and I coupled them by allowing certain metabolites to be transferred and to be accumulated. So in the plastic, we can store starch. So the starch can be stored and later on also retrieved from the plastic. And in the vacuole, we can store sucrose, carboxylic acids, nitrate, and amino acids. And while I'm saying this, so I'm not the person who invented this. George already pointed it out earlier. So this is really based on a deal modeling framework that Maurice developed a couple of years ago. For our project, we really just extended it from a 2-phase model to a 24 time step model. So 24 means one model for each hour of the day. And in our case study, we're looking at a mature leaf. So that means our optimization function is the export of SAP to the phloem. And I'll be referring to this as phloem output from now. And then instead of looking at metabolism at each individual time point, we're optimizing the whole 24-hour cycle. So we're trying to find an optimal solution for each hour. Well, we're trying to find an optimal solution for the whole day, but which then allows us to draw conclusions about the metabolic behavior at each individual hour of the day. So having this said, the model as it is now, if we were to run our simulations, we would actually obtain the very same behavior for each hour of the day. And this is because we haven't really included any external constraints. So this was then the second step of the modeling procedure, the addition of external constraints. So here I'm showing you that we included the ideal light curve. So this is an input constraint that went into our model. And we also constrained the day and night ratio of phloem output and for nitrogen uptake. And this was based on experimental measurements from the literature. So now putting this all together, we can run our simulations, and this is what I'm showing you here. So here you can see the hours of the day starting from sunrise, and here you can see in yellow the input light curve. So for this particular scenario, we're looking at a moderate light intensity of 250 micromole per square meter per second. And then in orange, you can see the flux through Rubisco, and in gray, you can see the CO2 uptake. And what you can see here is actually both fluxes, Rubisco and the CO2 uptake flux, they nicely follow the light curve. And then we can also look at the starch accumulation pattern. So we see that starch is being accumulated during the day, and then it's being degraded at night to sustain metabolism. So altogether, this is really what we consider C3 metabolism. So we were really happy to see this. But of course, we couldn't stop here because, I mean, for now, we've included light into our model, but we still neglected two of the main parameters that influence water loss through transpiration. That's the temperature and relative humidity. And here I'm showing you data that I'll be using throughout the simulations. So we're looking at an ambient temperature range between 50 and 30 degrees, and relative humidity ranges between 40 and 100%. Now, in order to include these two parameters, I started by looking at the gas diffusion equation. So this gas diffusion equation that describes gas exchange and water loss through the stomata. And I made a few simplifying assumptions, which then allowed us to relate the system's demand for CO2. So this is what you can see here. So this is the CO2 uptake. This is related to the water loss through the stomata. So this you can see here. And this relationship is only dependent on the temperature and the relative humidity. And also what is important to notice here is that this relationship is linear, and thus it keeps the overall optimization problem linear. To explain this maybe in a bit more visual way, we can look at this graphic here. So if we consider C3 metabolism during the day when temperature is high and relative humidity is low, if C3 plants open their stomata, CO2 can enter the system, but we also see at the same time a relatively high water loss. Then at nighttime, the stomata are closed and we see hardly any gas exchange. Now, the same holds true for CAM plants during the day. However, at night, when the temperatures are low and relative humidity is high, CAM plants open their stomata, CO2 can enter the system. And because of the external condition, we have relatively little water loss. So we can now combine these models, so we can couple the metabolic model to the system's demand for CO2, or rather to the water loss that is caused by the system's demand for CO2. And we can now ask the question, what happens if we apply water-saving constraints? And we can analyze this by performing a Pareto analysis. So Pareto analysis allows us to study the trade-off between two competing objectives. In our particular case, we're looking at productivity on the one hand and water saving on the other hand. And what I'll be showing you next is the transition. So we're really going to go along this Pareto frontier. So we will be decreasing the required phloem output. So we're decreasing productivity, but at the same time, we're putting more weight on water saving. So we are increasing water saving. And what happens when we move along this trajectory is the following. So you can see that CO2 uptake stops during the day, so that indicates that the stomata are being closed. And we see that CO2 uptake restarts at night. So the stomata open at night, and at the same time, we also see accumulation of carboxylic acids. So carboxylic acids are being accumulated at night, and then they're being degraded throughout the day. And of course, as we're moving along this frontier and as we're applying water saving constraints, this behavior becomes more pronounced. So now we can look at the actual Pareto frontier, and this is what I'm showing you here. So here we're looking at phloem output in percent of a C3 leaf and at water loss in percent of a C3 leaf. And what is important to notice here is that this relationship is not linear. So for example, if we're looking at 80% phloem output, we can see that we're not even using 50% of the water as compared to the C3 scenario. So that means that our model predicts that we can achieve a tremendous water saving while maintaining a very high productivity by partially switching to a CAM-like mechanism. And this 80% productivity I'll be using as a standard scenario for the future analysis that I'm showing you now. So before I will actually dive into the actual flux distributions, I want to highlight two more general observations. So one thing that I noticed when I looked at these plots is that the stomata only close for a relatively short period during the day, and they only reopen for a relatively short period during the night. And my first assumption was that this might be related to the vacuolar storage capacity, because the limit that we put on vacuolar storage capacity really was an approximation combining various factors such as leaf thickness, mesophyll conductance, and really a lot of factors that were estimated from the literature. So we thought that we might over or underestimate one of these values. So we decided to simply repeat the analysis with the difference that we're now lifting the constraint on vacuolar storage capacity. So on this side, on the left-hand side, you can see the same simulation that I just showed you. And on the right-hand side, we have an unlimited vacuolar storage capacity. And if we're now repeating the simulations, we see clear differences in the metabolic behavior. And here, most important thing that I want to point out is that this is a different axis. So this axis is actually twice as high as this axis. And what we can see if we go to our standard scenario, so standard scenario means 80% of the phloem output as compared to a C3 leaf, we see that with the unlimited constraints, the stomata remain open for an extended period at night, and they remain completely closed during the day. This is also reflected in the levels of carboxylic acid. So we have much higher levels of carboxylic acids accumulating at night and then being degraded during the day, but also the starch levels are much higher. So we do see metabolic differences, and this is then also reflected in the actual water saving potential. So we end up having 6% more water saving for the unlimited vacuolar storage capacity. Another thing that I want to point out is when I looked at these plots, they reminded me of some textbook representation of the 4 phases of the CAM cycle. So what are now these 4 phases? So each CAM plant goes through phase 3 and phase 1 of the CAM cycle. So phase 3 is when the stomata are closed during the day, and phase 1 is when the stomata reopen at nighttime to allow for gas exchange. But then some CAM plants go through what is referred to as phase 2 and 4 of the CAM cycle, and this is when the stomata open and reopen in the early hours of the day and also in the late afternoon hours of the day. And we can see that this behavior is quite prominent in our, what I call here, a CAM leaf scenario, whereas this behavior is much less prominent. We only observe the occurrence of phase 2 for the unlimited vacuolar storage capacity. So the take-home message here is that our model predicts that the limited vacuolar storage capacity really is a driver for the occurrence of phase 2 and phase 4 of the CAM cycle. Now, finally, in the last couple of minutes, I want to dig deeper into the actual flux distribution. And as we're looking at a 24-hour model, there's actually quite a lot to investigate. So I started this analysis by looking at those reactions that fix CO2 and release CO2. So again, here we're looking at the hours of the day. And on top, you see CO2 releasing reactions. On the bottom, you see CO2 fixing reactions. And it's no surprise, during the day, most of the CO2 is being fixed by Rubisco, what is shown in red here. However, when we looked at nighttime CO2 fixation, to our surprise, we had to realize that it's not only PEP carboxylase that is fixing CO2, but we also see mitochondrial isocitrate dehydrogenase fixing CO2. So this was a really interesting observation. And when we had a closer look at the storage metabolites and at the linker fluxes, it actually turned out that it's not malate, that is, or malic acid, that's the main C4 acid that is accumulating, but that in our model we actually see isocitrate or citrate accumulating at night and then being degraded during the day. And additionally to this, we also see an increased daytime accumulation of proline, and then proline is being degraded again during the course of the night. So this is the particular enzyme that I was talking about, isocitrate dehydrogenase, and George already pointed this out earlier. So this enzyme was believed to be irreversible and to only operate in the CO2 releasing reaction, but it was actually shown in MFA studies in developing rapeseed and soybean embryos that this reaction can indeed operate backwards and fix CO2. And this is also what we see in our model predictions. So in the end, when we put all this information together, we had to extend our classic CAM cycle by an additional cycle. So what our model predicts is that isocitrate that was stored at night is released from the vacuole during the day. It releases CO2, which then can enter the Calvin-Benson cycle and be used for starch synthesis. And the resulting 2-oxoglutarate is then converted into proline by a series of reactions. And then proline is released at night and can serve as a precursor in the mitochondria to generate 2-oxoglutarate, which then serves as a precursor in the TCA cycle to fix CO2 and to build up new citrate or isocitrate, which then can close the cycle and be stored in the vacuole again. One last thing that I want to point out, we found this actually very, very interesting, these observations. And we were curious to see if there's any evidence for this behavior to occur in the literature. And we came across a study from the 90s where the authors investigated proline accumulation in ice plants. So ice plant is one of these facultative CAM plants that can switch from C3 to CAM under inductive conditions. And what the authors observed under these conditions is that proline was accumulating during the day and being degraded at night in a very particular pattern. And what we think what could actually happen in the system is that in a similar way as our model predicts, daytime proline accumulation really serves as a precursor for carbon fixation at night through mitochondrial isocitrate dehydrogenase. And then once the CAM induction and mostly PEP carboxylase levels have reached their full CAM cycle, then the thermodynamically more favorable enzymes, so PEP carboxylase, might actually take over. For reasons of time, I'll be skipping the analysis of the environmental parameters, and I'll just tell you about the main findings and jump straight to the summary. So what I showed you in this presentation is that the vacuolar storage capacity limits water saving and also influences the extent of phases 2 and 4 of the CAM cycle. We've seen that our model proposes an alternative CAM cycle or alternative CAM-like flux modes, which indeed can contribute to water saving. So most of all, this includes nighttime carbon fixation by isocitrate dehydrogenase. And last but not least, I haven't showed you this, but I can tell you that our analysis of the environmental parameters showed that especially under low light conditions and under short day conditions, the additional carbon fixation by isocitrate dehydrogenase is of advantage. And of course, this makes it a very interesting engineering target for crops that are grown in temperate climates. So having this said, I want to come to the last slide and to the acknowledgement. And I really would like to thank everyone from the Department of Plant Sciences and particularly all the members of the Sweetlove group that really made my time in Oxford really unique and really enjoyable. And then specifically for special thanks to Lee Sweetlove, George, and my colleagues Sanu, Thomas, and Noah. And having this said, I would also like to thank you for your attention.
Thank you very much, Nadine. That was an excellent talk. There may be a few questions. I'm just looking in the chat now. We'll see what comes through. There was a question during your more introductory material where someone, I'm just reading it now, Ashutosh Sanu said, what are the modifications we need to implement in FBA equations when we're working on dynamic fluxes? Now, in fact, I think you've illustrated to some extent how one can approach a dynamic process by FBA with your own work. But would you like to comment further on the notion of dynamic FBA?
So I didn't quite hear the first part of your question. What was the exact question?
Well, Ashutosh Sanu said, what are the modifications we need to implement in FBA equations when we're working on dynamic fluxes? Now, I think this question was triggered by some of your comments in the early part of your talk. But of course, you went on to illustrate a dynamic situation. So maybe it would be helpful to say just a little bit more about the whole concept of dynamic FBA.
Yeah, maybe it's worth pointing out that what I was showing here, and also I think all of the other approaches that really focus on modeling deal metabolism that we're going to hear today are not to be mistaken with dynamic FBA in its classic sense. So these are really two different approaches. So what we are doing is still FBA, but we're using FBA to simulate dynamic behavior, and our dynamics really come from the fact that we're allowing certain metabolites to accumulate and to be transferred from one time point to another. This really has the advantage that we can still look at really large networks. So our models are still linear and can be resolved in basically no time. Whereas if we're looking at what is actually referred to by dynamic FBA, which then also splits into two different variants, this typically involves also ODEs, so ordinary differential equations, which are much more difficult to solve, and which in a way still limit upscaling FBA or dynamic FBA approaches to really large scale networks, because they're still computationally quite challenging and also involve knowledge about kinetics that are involved. So I just wanted to point out that there needs to be distinguished between these two approaches.
Yeah, that's very helpful. I think that will guide the person who set the question. Any other questions? Well, Poonam Jyoti asked a rather general question again about what prerequisites should we consider for FBA. A very general question, which of course you've illustrated in your particular case. Have you any further comments on the way one should set about doing FBA?
Well, I could start talking about it, but I think we're a little bit short on time already, and I really would like to hand this question over to all the speakers that come next, and maybe also to the person who asked the question, to really try to get your question by listening to the upcoming talks, because I think it will be much more informative to hear about very specific examples than just listening to me giving some general statements. I think it's really helpful to talk about the very specific examples, because they can be really illustrative.
Yes, okay. I'm just looking down. Don't worry about the time, because unfortunately Tarit or Man's talk has had to be cancelled, I believe, because he's not well. So we do have a bit of a gap in the schedule. There seem to be some more questions coming through, or no, people answering questions, other people's questions. Maurice is active. Right, so here's a question.
So if I could maybe make a quick comment, because I saw these questions popping up while I was speaking. I personally would have preferred not to see all these chats and conversations going on. So maybe that's just a hint to make it a bit easier for the next speakers. So maybe try to have your questions in mind, but discuss them towards the end of the presentation. At least I felt a little bit distracted.
Ah, right, yes. Okay, well, I'm trying to locate I'm trying to distinguish answers from Maurice to people to questions. Oh, here's one. Chandrakant Joshi says, how detailed our metabolic model should be in FBA? Does the model also need metabolic regulation information?
Well, that's a very crucial point, I think, that he's touching here, because, I mean, the FBA models, as we're using them, don't really include any regulatory events. And I think that there are methods to include regulatory events, but I think it's really the beauty and the elegance of the approach that we don't necessarily have to put in these information, or rather assumptions, because we see that if our objective function is wisely chosen, we are able to simulate plant metabolism without actually to explicitly account for these regulatory events. So,
yeah, thank you. And then there's another question from Poonam. Does the objective function differ across DL cycle times? How do we decide on objective functions?
well, that's also one of the most crucial questions, right? I think Maurice might also be able to comment on this later, because I think he has reviewed the choice of the objective function. But yeah, I mean, the objective function really depends on the system that you're looking at. So first of all, for example, if we're looking at a growing leaf, or if we're looking at a mature leaf, or if we're looking at a source organ or a sink organ. And so the other point that I wanted to make with respect to the objective function, so this is something that should be crucially analyzed, and analyzed also in terms of what happens if we change our objective function. But what is also really important here is the inclusion of maintenance cost. And again, I would like to point to Maurice's work, and maybe he can comment on this, because he showed that the inclusion of cellular maintenance is also a really, really important point when it comes to having accurate predictions about cellular metabolism. So it's really two crucial parameters, or two crucial inputs or assumptions that we need to consider.
Thank you. There's a question from Yogesh Pand. How can we validate FBA models, especially the water-saving model?
Yeah. So let me start with a specific answer. So the water-saving model, we validated it by looking at the literature and by comparing the water loss predictions that we obtained from our model. We compared this to average transpiration rates from the literature, and we saw that our model is very much in line with what is known from the literature. So this was the very specific validation for our model. Well, if you want to look at it from this perspective, the fact that we do see C3 metabolism over the DL cycle in a way is also a validation of the model, because it shows that we can actually recapture the behavior that is known from a very minimally constrained model. So our model really has very few input parameters. And then to give a more general answer to this question, so the first thing, of course, is to compare our model prediction to what is known from the literature, and to compare it also, and I think you made a point in the beginning to compare it to measurements that we have from metabolic flux analyses. And as George pointed out earlier, there might actually be discrepancies, but then again, analyzing these discrepancies can lead to very interesting biological observations.
Okay, there are a couple of questions from Maurice. The first one says, I assume the 24 looped, the hour 24 looped back to hour 1, and all 24 hours were simulated as a single optimization problem. Is that correct?
Yeah, I don't think this needs much comment, right?
Yes, exactly.
That's correct.
And if I just scroll down the chat, there is another question from him, but I lost it at the moment. But Maurice is actually online, so I don't know whether you want to ask that question yourself, Maurice. It's about the vacuole, but the chat is very
okay, might as well. Can people hear me?
Yes,
Okay.
Maurice, ask your question, because I've found it now without vacuolar storage, but you ask it.
Sure. Yeah, I was just wondering, how was the vacuolar storage capacity modeled? Should I actually wait for Sanu's talk, or maybe you want to give a hint of how did you do that?
Actually, the way the vacuolar storage capacity was modeled, it's very simple. We simply put an upper limit to the linker flux, because I mean, you notice yourself, the linker flux can be used to also refer back to actual concentrations if you have knowledge about the volume of your respective compartment. So we basically went backwards. We had knowledge about the size of the vacuole and about the storage capacity. We started actually by looking at malate, and based on this, we calculated an upper limit for the linker flux.
Okay, thank you.
You're welcome.
Thanks, Maurice. There's a very specific question from Brendan referring to slide 91. So he says, why does the model create proline from 2-oxoglutarate, then convert it back to 2-oxoglutarate rather than just storing glutamate or 2-oxoglutarate?
Yeah, that's a good question. Actually, I remember that I was asking myself the very same question, because intuitively it would be a shortcut just to say to store 2-oxoglutarate in the vacuole. I don't, to be very honest, I don't remember the exact answer from the top of my head, but I can imagine that it might be somehow related also to energetics, so that it might be more energetically more advantageous to have certain reaction steps occurring during the day and then reversing them at night. But yeah, really, I can't give you the very exact answer from the top of my head now.
Okay, I think we're probably running out of time. There are a few more questions in the chat, but you can probably see those, Nadine. So at some point you might want to respond to them directly. We probably do need to close the discussion at this point so that we can get on to the next talk.
Yeah. I just wanted to make, I saw Sanu's comments that we don't have 2-oxoglutarate in the vacuole, but I actually tested and allowed for 2-oxoglutarate, and proline was still the preferred compound to be stored.
Good point. Yeah. Okay, so I think we'll draw this discussion to a close, but Nadine, if you can pop answers in the chat to the 2 or 3 questions that we've not had time for, that would be great. Otherwise, thank you very much. An excellent talk, and some good questions came in there. And I think at that point, I hand back to, is it to Chandrakant to set up the next talk?
Yes, George. Chandrakant.
And just before I start this, Shyam, how long has Sanu got to talk?
About 30 plus 5 minutes.
30 plus 5, okay, which is what I think Sanu was expecting. Okay, so let me welcome Sanu, who, as you can see from the biography, is a metabolic modeler from Kerala. And he's currently working with Lee Sweetlove and me in Oxford. He's been working on plant metabolism now for quite a number of years. He moved to Oxford several years ago. Prior to that, he received his PhD from the University of Toulouse, where he was working on a rather different system, a trypanosome system. So Sanu is going to talk to us about a specialized form of flux balance analysis, which is being dubbed GROW FBA. So over to you, Sanu.
Thank you, George. See if I can share my screen. Is my screen visible?
You're not very audible.
Oh, can you see my screen?
I can see your screen, but you're very, very quiet.
Okay, I'll speak up.
You certainly will need to, I'm afraid.
Okay. All right. Hello, everyone. So in the last 2 talks, you heard about FBA and how it's being used to model metabolism. In this talk, I'll be focusing on using FBA to model osmotic cell expansion. And I'll be using the case of developing tomato fruits as an example to highlight this issue. Right. So traditionally, FBA models growth by identifying a flux distribution that supports the accumulation of biomass. Right. So you do this by first estimating the biomass composition of your tissue sample. Then you add a reaction to your stoichiometric matrix where you say, I would like to drain the biomass elements in a particular ratio. You then set this as the objective of your model, and then say, I would like to perform FBA, or to be more precise, you do parsimonious FBA, because that also minimizes the sum of fluxes, which in a way is trying to identify an efficient flux map, an efficient flux distribution that still maximizes your objective. So you can then use additional flux constraints to improve your flux prediction, and then you will see that it can get closer to what real fluxes look like. But essentially what's happening here is the multiplication of biomass, which is essentially growth by cell division. Right, but growth in plants is not all division. In fact, cell expansion is responsible for most of the observable growth. If you look at leaves, for example, you'll see that the cell division phase, which is shown in green here, is dominant during the early stages of development. And you'll see that it soon transitions to a system where cell expansion is dominant. This is the same case in, say, developing tomato fruits. You'll see that the early stages, you have cell division being dominant. And then as cell division drops up, cell expansion takes over. And then in the case of a fruit, you also have this ripening stage where you go from green to red. Now, metabolic fluxes reflect the demands of the system, right? And to study growth, we need to, because cell expansion is responsible for so much of the observable growth, to study fluxes during growth, we need to model expansion. And so in this talk, I'll be looking at how fluxes are different between expanding and dividing systems. And we'll also be focusing on a bit more of a practical application of this approach in modeling tomato fruits. So classic modeling, which is kinetic modeling, can account for changes to cell volume or volume of subcellular compartments as well. And they have been used to model metabolism in developing tomato fruits that go through expansion phase. So here's a model that does that. And if you look at this particular reaction here, which is this particular metabolite here, which is represented by this equation, you can see that the volume of the plastid, which is a subcellular compartment that metabolite is in, is taken into account during the modeling. However, kinetic models can only cover small parts of metabolism because they rely so much on enzyme kinetic parameters and metabolite and enzyme concentrations, and this data is difficult to get by. Another problem with kinetic models is that they essentially form nonlinear problems, which soon become computationally expensive the larger it gets. And this is where FBA comes in. So FBA assumes metabolism is at steady state, and in which case all the fluxes can be associated with each other using linear equations. However, because of this simplification of the system, the model is no longer sensitive to changes in volume. And so we developed a new formulation to model growth by osmotic expansion using FBA. We're calling it a Grow FBA, and I'll sort of explain how it's done. And there will be a lot of equations here, but the reason I'm showing this equation is just to show you how simple it is. It's actually not that complicated if you look at how these equations were built up. Now, cell expansion is a result of accumulation of osmolytes, right? Over here. And then followed by the influx of water, and your cells expand. Now, FBA is not sensitive to changes in volume, but it can account for the amount of metabolites and hence the osmoticum. So if you write down the equation of the osmolarity of the cell in terms of its molar concentration, you have this term here, which I'll be simplifying. So this is the osmotic coefficient, which is a property of the solution that characterizes its deviation from ideal behavior. If we assume an ideal solution and an ideal behavior, we can replace we can say phi equals 1. And so you can simplify this equation. And then we can replace molar concentration by mass by volume. The number of moles per volume. And rearrange this equation, and we get this equation. So according to this equation, if we know the volume of the cell and its osmolarity, we can introduce constraints to limit the content of the cell. And let's call this equation 1. We'll be using it a few slides later. So equation 1 can constrain how much metabolites can accumulate in a cell of a given size. But how much of that accumulated metabolites is vacuolar? So for this, we need another equation. And so for the derivation, let's look at the steady state scenario between the vacuolar and the cytoplasmic space. At steady state, the water potential between the vacuolar space and the cytoplasmic space is the same. And the water potential can be represented in terms of turgor pressure and osmotic pressure. And if we replace this in the equation, we basically get turgor pressure of the vacuole minus osmotic pressure of the vacuole equals the turgor pressure of the cytoplasmic space minus osmotic pressure of the cytoplasmic space. Since the turgor pressure of the vacuole and turgor pressure of the cytosol is being offered by the same tonoplasm membrane, at steady state, they have to be equal. And so you get your equation now becomes a lot simpler. And from Van Hoff's reaction, we can replace the term osmotic pressure with RTC, where R is the gas constant, T is temperature in Kelvin, and C is solute concentration. And in an ideal system, we can assume the osmolality equals the osmolarity. So if we cancel out the R and T, we have the simpler equation again. And from assuming ideal behavior, we can replace it with the Van Hoff factor into the molar concentration, the same equation we used in the last slide. And again, replacing molar concentration with number of moles by volume, we get this equation, and rearrange the equation, and we get this equation here. So what this equation basically says is that if you know the ratio of the volume of the vacuolar space to the cytoplasmic space, we could use that to set the amount of metabolites that would accumulate in the vacuole to the amount of metabolites that would accumulate, well, osmolytes to be more specific, the amount of osmolytes that would accumulate in the cytoplasmic space. And this would be our equation 2. Now, how would we apply this equation to our model? Well, before this, I can further simplify this because of the nature of my model. So the Van Hoff factor is the ratio between the number of particles formed when a solute is dissolved in a solution and the number of moles of a solute dissolved. For example, in an ideal solution, you take 1 mole of sodium chloride and you dissolve it, you get 1 mole of sodium and 1 mole of chloride. And so the Van Hoff factor is 2. But for sucrose, it doesn't dissociate if it's a very dilute solution, and so you get 1. Now, in the model that I'm working with, all the metabolites are already presented in their dissociated states. So I can replace the Van Hoff factor with 1, and I have the simplified equation 1 and equation 2. And I'll show you how one can easily apply this to a model that basically changes cell volume. So let's take the system where the model changes from an initial cell volume to a final cell volume. So you have an initial metabolite content that matches the cell volume, and then you have a final metabolite content. Now, you can make up the osmolytes in the final metabolite content by just retaining the osmolytes in the initial metabolite. Initial cell content. Or you can import new metabolites and synthesize new osmolytes for the final cell composition. Anyway, the idea is that anytime an osmolyte accumulates in the cytosol, you would add a pseudometabolite, a fake metabolite, that would represent the osmotic contribution of that metabolite to the cytosol. And anytime a metabolite would accumulate in the vacuole, we would add a pseudometabolite or a fake metabolite that would represent the osmotic contribution of that metabolite to the vacuole. So we have these 2 pseudometabolites, and if we add a reaction to our model in this manner, we're essentially satisfying the equation 2 constraint. And if we set the flux of that reaction to V cell as a product of V cell and C cell, we're actually satisfying the constraint represented by equation 1. So in a very simple manner, we're able to apply osmotic constraints to the model. However, that's not just what an expanding cell needs. It also has structural demands. As cell expands, it needs to synthesize cell wall. And we wanted to try and stay away from experimental data as much as possible. And so we assumed the cell is cubic in shape with an edge of A, and the cell wall is evenly thick with a thickness of B. And we calculated so if you consider the volume of the cell wall as the volume of the total cell minus the cell without the cell wall, you can easily represent that in this equation. And so the volume of the cell can be represented in terms of the volume of the cell wall can be represented in terms of volume of the cell. And you can also represent, if you assume that the cell wall is composed of only cellulose, and because in our model cellulose is represented as glucose units, we can estimate the number of moles of cellulose for a given cell wall, volume of cell wall, which is a function of the volume of the cell. And so we can estimate, if for a given cell volume, we can estimate the amount of cellulose that would be required. So we made some predictions, and we compared that to tomato pericarp data. And we were happy to see that these predictions were close to what was experimentally measured. And so we've moved on to the phospholipid demand that the cell will require to support that cell expansion. Again, we use the same assumption scheme. So you would assume your cell is a cube, you would have an evenly thick cell wall, and now additionally you have an evenly thick cell membrane. Again, in a similar fashion, we generated an equation where the volume of the cell membrane is a function of the volume of the cell. And again, in the similar fashion to what was done before, we found an equation where we could estimate the moles of cell membrane phospholipids as a function of the volume of the cell. Now, if FPM is the fraction of the cell's total phospholipid that is associated with cell membrane, then if we introduce this term into the denominator here, we could estimate the moles of phospholipid that would need to satisfy a particular cell volume. And again, we compared this to our measured data in tomato pericarp, and we found that it matches quite well. Now that cell wall and phospholipids are done, the next cell main biomass demand is protein. So we know that to maintain an unhampered cell function, as the cells expand, they need to maintain an optimal protein concentration. So we made an assumption to simplify things that the vacuolar fraction of cell protein is negligible. And so we can represent the moles of protein as the molar concentration of protein into the volume of the cytoplasmic space. Now, I know the term moles of protein can be confusing. It's simply based on how the reaction for generating a protein is coded in the model. So again, we wanted to see if this equation makes reasonable predictions. And so we compared that to our tomato pericarp data. And it actually fits much better than the previous two equations. So we're happy that by using these equations, we can avoid actually using experimental data and stick to using basically one or two parameters, which in this case is the volume of the cell. Right, so now that we had the osmotic and structural constraints for expanding cells, we decided to model cells during expansion. So we model cells that are expanding and accumulating only organic solutes and only inorganic solutes. We also modeled dividing cells using traditional FBA based on the conditions that was reported in the study. And as you can see from the flux distributions, they are completely different. Well, they do accumulate some of the same things, but the amount of flux going to these demands are very different. In fact, if you look at the energy expenditure, the total amount of energy the cells spend is similar within the same range. However, what they invest in is very different, especially the expanding cells compared to the dividing cells, because the expanding cells invest so much of their energy into gathering osmolytes, whereas the dividing cells invest more in structural biomass elements, like cell wall, protein, and phospholipids. Now, moving on to modeling a developing tomato fruit. Now, this tomato fruit at DPA 8 has to go through multiple developmental stages to get to a ripe tomato fruit at 59 days post-anthesis. So we decided to split this up into 10 different developmental stages. And an approach that is very similar to what Nadine did, we basically created a copy of a model 10 times to represent the different developmental substages, and then connected those models together. So this particular reaction that represents the movement of starch from a phase to the subsequent phase can represent the accumulation and transfer of starch from a time point to a different time point, from a developmental stage to the next developmental stage. We then permitted an initial level of metabolites to the model, which would represent the metabolic composition of the small fruit here. And then we constrain the final composition of the ripe tomato fruit. We provided access to the phloem for all these models, and then also provided it access to xylem where it could take up ions and water. And we also set up basically options for the model to accumulate or degrade biomass elements based on the constraints that we estimated earlier. And we needed more constraints to actually set the model up properly. So we know that as metabolites accumulate, vacuolar pH is likely perturbed, and we found evidence for this in cherry tomato. So we adapted this data to set the vacuolar pH of our tomato fruit for the different developmental stages. You can see that here. And as Nadine mentioned, non-growth associated maintenance is a very important cellular demand in any metabolic system. So, and this is something that cannot be predicted by FBA models. So we found maintenance respiration rates reported in literature, and we set up maintenance costs based on this respiration rate. Now, finally, we had a model that we can actually run FBA on. So we performed FBA, and with the objective of maximizing the fruit organic content.
Sanu, we've lost you.
The expanding cell. And while there were some changes, which you can see.
Sanu, could I just interrupt?
Yeah, sure, George.
We lost you at the beginning of this slide. Could you go back to the beginning of it and start from that point?
Sure. So,
yeah.
Okay.
Great.
While after constraining maintenance, we were a bit more confident that we could run an FBA and get reasonably good fluxes out of the model. So we performed FBA with the objective of maximizing fruit organic content. And what we found was, well, it wasn't surprisingly, what we found was a very large fruit, about 200 times the size of a normal fruit. However, when we added osmotic constraints into the model, what we got was a fruit that was very similar to a normal fruit in size, in organic content. And so, in other words, when we added grow FBA constraints, it significantly improved our developing organ model. And we also added the biomass constraints, the cellulose, the phospholipid, and the protein demand constraints. And there were a few changes. If you look at this part of the osmoticon, you can see that there are a few changes in the middle of the developmental process. However, it was not very significant. We suspected this was because at this point the model has unlimited access to phloem, and in the literature we see that the carbon uptake decreases as the fruit size increases. So we had to introduce additional constraints on the model, which would limit the amount of phloem based on how big the fruit is getting. And interestingly, what we found was that when we introduced these constraints, we could see starch being accumulated. So at this point, we decided it's time to evaluate the quality of our model. So we looked at the model. So these are the model predictions in gray here. And in white here are estimations based on measured organic content in fruits. So what you can see here is that the model is able to predict glucose, fructose, glutamine, and glutamate levels reasonably well. And these comprise like 89% of the organic soluble content in fruits. In other words, it suggests that the accumulation patterns represented here is an efficient way to drive osmotic expansion. There were other metabolites that the model was unable to predict a good pattern for example, GABA. And it suggests that the accumulation of these metabolites may be involved in other aspects of fruit physiology, for example, taste, texture, insect resistant, etc. Now, like George mentioned, these models should also give you a biological insight to actually make this process worth pursuing. And so so we look back into the interesting things we found from the model. And one of the things that we found out was that, which was mentioned earlier, is that when phloem uptake constraints were introduced in the model, there was an appearance of transitory starch stores. So this is starch appearing in the beginning, and then starch disappears towards the end, towards fruit ripening. And this accumulation of starch during the early stages and then disappearance before ripening is well documented in tomato fruits. So we looked at what was the status of the model before the phloem uptake is being introduced and after it is being introduced. So the dotted line you see here is the phloem uptake constraint. And as you can see here, it's at 1,000. It's like the default maximum phloem uptake. So there is no uptake rate constraint. And here you can see that there is a dotted line which is limiting the phloem uptake rate. So, and then what we did was we applied this phloem uptake limit that we were going to apply, that we hadn't applied yet, onto this plot. And what you'll see is that there are certain parts of the developmental stage where the phloem uptake is above the limit. And if we trace back to exactly what part of expansion phase this happens, we'll see that this happens around when the expansion rate is maximal and until the end of the expansion phase. So going back to the system where we've already applied the constraint, if you look at how the different carbohydrate storage has changed, you can see that before the constraints were applied, there was no starch. And after the constraints were applied, you would see starch buildup. But in the region where the phloem was above the limit, which is this region here, you would see starch being degraded. In other words, the starch accumulation seems to be associated with the increased demand during cell expansion, which cannot be met by the phloem alone. So another thing we tried was, in this scenario where there is a phloem uptake constraint, we've told the model you cannot accumulate starch. And the result was the fruit organic content was much lower, about less than 40%. So what does this tell you? Well, fruit initiation in tomato trusses is staggered. So in other words, you have along the truss, you have tomatoes of different size that are growing. Now, if the metabolism is even in all the tomatoes, what you expect to happen is the bigger tomato would have a bigger sink strength and the smaller tomato will have a smaller sink strength. And if this continues, what you would end up is one big tomato and a whole bunch of small tomatoes. However, if starch synthesis is triggered during early stages of development, that would increase the sink strength of the smaller fruits. And this, coupled with the fact that when the starch is degraded, when the tomatoes approach mature green stage, that would decrease the sink strength of the larger fruits. And the end result would be that you would have a truss with even-sized tomatoes. Now, that's one of the biological insights, but there is, as with all modeling approaches, this modeling approach has its positive and negative effects. But let's discuss the positives first. One of the biggest thing is that metabolism is minimally constrained. So the added advantage of having a metabolism that is minimally constrained is that when there is a flux prediction, you don't see just what is happening. You see why. You can find out why it's happening. So for example, in the case of starch degradation, we were able to find out that, oh, the starch degradation is happening because, why the starch accumulated. We found that in the later stages, the demand for carbon is higher than what is being allowed because of the phloem uptake rate. So because of the metabolism is minimally constrained, the fluxes you get out of it, we can find an explanation for it. And it is likely that you may be able to draw biological insights from this. Another upside about the model is that the GRFBA imposes osmotic demand on the metabolic system. And as you saw, that actually helped improve the metabolic content prediction. So you can see that during the different developmental stages, we were able to predict the bulk of the osmoticum with reasonably well accuracy. Finally, this model uses the multi-phase approach that Maurice invented for us. So the multi-phase nature and single optimization nature of the problem permits the adjustments of metabolic fluxes during the early stages of development to meet the demands of the later stages. For example, this goes back to the starch problem, where in the later stages, because in the early stages, the starch accumulation occurred because during the later stages, the cell would not be able to meet its demand from phloem alone. Now, going to the downside, the model is still an FBA model. So it is unable to account for futile cycles. It does not account for regulation or signaling. However, there are kinetic models on fruit metabolism that does this. So if you find a way to couple the kinetic models with our FBA models, we may be able to build a hybrid model that can account for futile cycles and can account for regulation, signaling, enzyme kinetics, and so on. And then there's another challenge with this model, that it does not impose constraints based on insect resistance, texture, and flavor. However, this is very difficult, but maybe not impossible. And I think if it is possible, it will be very relevant, especially given the interest of metabolites such as GABA has in understanding biology. So in this talk, I've talked about how GrowFBA can be used to model developing tomato fruits. However, you can use this to model any developing organ, because like I showed earlier, expansion phase is responsible for much of the observable growth in plants. So for example, in the case of the area of elongation in roots, you could use GrowFBA to try and get meaningful insights into metabolism. Another example where you can use GrowFBA is in cells that would change their volume. For example, God cells during the opening and closing of stomata. And so to summarize what I discussed in this talk, osmotic expansion is responsible for most of the observable growth. And we've developed a GrowFBA framework to capture this. We used the GrowFBA constraints and our multi-phase modeling approach to develop a model of developing tomato fruits. And thanks to the osmotic constraints, we were able to predict reasonably well fruit organic content throughout the developmental stages. And then finally, thanks to the osmotic constraints and the multi-phase modeling approach, we were able to have generate a hypothesis for the role of transitory starch in tomatoes. And this work obviously the credit of this work goes to Lee and George, who supervised this work, and of course to our collaborators at the Max Planck Institute of Molecular Plant Physiology, especially Jose, who's a postdoc there, who did all the experimental work and generated tons of data for me to use. And finally, I'd like to thank Ericast, but finally, I'd like to thank the audience for listening in to my rambling. Thank you very much.
Thank you very much, Sanu. You might want to turn on your video so that people can see you.
Oh, I didn't realize my video is off. That's a lot of hand gestures that were wasted.
Anyway, there's time for some questions, and the first question comes from Rahul Shaw, and he says, can this approach be used to model endoduplicational polyploidy level with cell expansion? I'm not quite sure how to read that at the moment, but he wants to model reduplication or polyploidy level with cell expansion.
I'm not at the moment. I'm not really sure what that entails. Maybe we can discuss this at some point.
Yeah, I think this might be a question to take offline and explore precisely what Raoul's got in mind here. It looks like a complicated area. I'm waiting for other questions. Nothing much has come through yet. And you can see them as well, Sanu, but nothing is.
Yeah. I can see another question by Shakar Hill.
Oh, yes, there's one coming now. Grape has rather even size within a cluster, but they do not accumulate starch. Any explanations?
I think we would have to model that.
I think we would.
Yeah. The network's really complicated. I don't think would be fair for me to make assumptions.
That was a good point, but certainly something would need to be modeled. Any other questions? Ed Smith has come through. What limits the phloem uptake rate? Is it due to the other fruits developing on the same truss?
So all we can tell from the modeling is that the phloem uptake rate is related to the starch appearance of starch, transitory starch stores. We don't know what's triggering what. And I think it is likely that the starch would be triggering the phloem uptake rate, by manipulating the zinc strength. So you would starch degradation. So for example, during early stages of development, you have starch synthesis, which would drain sucrose and reduce the sucrose concentration inside the fruit. And so you would have a higher zinc strength. And when the fruits are bigger, at mature green phase, when the starch starts degrading, that provides additional sucrose and so decreases the concentration of, increases the concentration of sucrose in the mature green fruits, and so decreasing strength. But this is just a hypothesis. There is studies where they've, I think it's where they've played around with AGPAs and extended the starch accumulation phase. And I think they've sort of presented a similar idea that the starch may be responsible for manipulating zinc strength.
Okay, so there's a question in from Manish who says the fruit growth model is awesome, so that's very pleasing, Sanu. Does your objectives keep changing along with the grow parameter assumptions? So it's slightly difficult to know precisely what to take from that, but what's your answer?
So the objective of the model, so if I can go back to my slides and to that. So essentially the objective is to maximize the final metabolite composition, so the final organic content. That is the objective of it's a system where all the phases are modeled together to achieve the maximal organic content. So the grow FBA constraints are additional rules that the model has to follow. Rules that states it has to accumulate a certain amount of osmolytes to so that it matches the volume of the cell at that time point.
Okay, moving on. There's a question from Bertrand Gautier. Could you challenge your model by using data from transporter mutants in tomato affecting fruit size?
I think so. What do you have any particular transporters in mind? I think it depends on like how specific the transporters are, right? So if you want to model a specific transporter being knocked out, I think that should be possible. However, I think the model, I think it would be too early to tell because the model, you see, it depends on, it's not constrained very well. So it would try to find other avenues. Like for example, if you don't allow blue hexose uptake, it will try to take up sucrose. And if you don't allow sucrose uptake, I think it would try to take up hexose, but then it won't. There would be added costs, so it would probably accumulate more starch. But like I said, you have to test this. It's very difficult to predict what would happen because of the number of stages we're modeling here. We're modeling 10 different models put together, and each of these models have different constraints on them. So it's very difficult to predict without actually, so you would need to run this, I guess. And also you would need to know exactly what transporters you're trying to disable or restrict or, constrain, that sort of thing.
Okay, a question from Zanru Dai, who says, for the assumption turgor vac equals turgor site, so this is going back to your basic model, could you please explain further? I think there's a role of the cell wall for turgor.
Yes, so cell wall, so usually to get turgor pressure, you need resistance. So let me try and find that equation. So, right, so that's how cell wall offers resistance. Cell wall offers resistance. That's why when you mention turgor pressure, you automatically go to cell wall. But in a system where there is resistance from both sides, your tonoplast should be in the center. So the tonoplast should be offering resistance just because of the osmotic pressure on the other side. So, and because this is the same, the tonoplast is the same membrane that's being referred to here, they should have the same turgor pressure.
Yeah, okay. A couple of questions from Maurice. First one is how the stages connected to each other, and were the stages modeled in a single optimization or more like dynamic FBA?
The stages are all connected. So the starch, all the starches are connected. So the starch from one phase can go to this, can become the starch of the next phase, and so on. And it's a single optimization problem. So very similar to what Nadine did.
Yes, and his follow-up question is, and what are the pros and cons between single optimization versus dynamic FBA for this system?
Right. Well, for at least in this system, I think I'm happy that I went with the single optimization problem, because if I didn't go for the single optimization problem, I wouldn't be able to see the relationship between starch and film uptake constraint. So with dynamic FBA, your past can influence your future, but your future cannot influence your past. Whereas with the single optimization problem, your future can influence your past. So in terms of speaking with what's happening in nature, over the course of evolution, the system sort of optimizes to sort of predict what is going to happen. And then so it sort of prepares for it. I guess you could say this is the same as when the leaf starts to degrade starch, predicting the end of the night period. So something like that. So I think I am happy that I went with the single optimization problem.
Okay, Maurice seems to be satisfied with that, so that's all right. Zenru has come back again. What will happen if you modify the imported sugar from sucrose to hexose to represent the shift of phloem unloading pathways between symplastic and apoplastic paths? You've probably not tried that.
Yeah, I'm not sure. I haven't tried this.
Yeah, I mean, it's unknowable. One would have to run the model to find out. Yogesh Pant comes in with why pseudometabolites are used, and doesn't this influence the outcome of the model? Could you say a little bit more about the pseudometabolite concept, please?
Right. So the pseudometabolites are essentially fake metabolite. They shouldn't have a formula. They shouldn't have so it's just something it's just something that I use. So you can get around using it. You don't have to use it. There are something that you can have flux ratio constraints. So I've been using Cobra Pi, and Cobra Pi lets you do constraints. I use pseudometabolites because then I can see what is happening. So I can see the you know, how the fluxes behave between. So all the fluxes that have the pseudometabolites, I can trace the pseudometabolites like I would trace the metabolite. And they don't influence the model because I make sure that the reactions involving them. So basically here you can see that when this is a normal reaction would be like that, all you're adding is a pseudometabolite, which you're draining away from the system. So they don't affect any other part of the stoichiometric matrix. All they're involved in is this particular reaction and that particular reaction. So yeah. So they shouldn't influence the outcome of the model. They would influence the outcome of the model because of this constraints that we put in. However, if we did not add this reaction and we were draining out the pseudometabolites on their own without this reaction, without these constraints here, then they would essentially have what you would get is the exact same solution if you didn't have a pseudometabolite.
Okay, and one final question from Mazia. Can this multi-phase modeling be used to model growth phases, that is lag, log, and stationary phases, of the plant cell division?
So for cell division, I think so the whole concept of multi-phase modeling. I think it should be possible, although I'm not sure what advantage we would have. I mean, we would have the advantage of, for example, if you would come under nutrient. For example, you're having an organ that's growing, and there is limited access to uptake. I guess then the multi-phase modeling would help. To be honest, I'm not sure. I think we would have to try this. I haven't quite thought of trying this because how traditional FBA is effective in modeling cell division. So honestly, I haven't thought about this, but it's a good question. I think it's worth a try because it's given how fast and efficient FBA is. I think it's worth doing a try.
Yeah. Okay, good. I think that brings us to the end of the question. So thank you very much again, Sanu, for an excellent talk.
George, can I make a final point?
Yes.
So if people manage to find the paper so you just have to Google grow FBA, there should be something called a binder link in the GitHub repository associated with this paper. If you go in there, you can basically do the modeling yourself. All the scripts, all the environment, everything's there. All you would have to do is keep pressing enter, and you would see metabolic fluxes being predicted in number form, not in graphs. So you can try things out yourself there.
Sounds like a good thing. Okay, so yes, thank you again. And at this point, I'll hand back to Shyam, who will tell us when we're going to resume.
Excellent session. Thanks. Thanks, George. Thanks, Nadine, Sanu. I think we had very insightful talks as well as nice discussions going on. And the final point about the binder link as well, I hope the audience could pick it up and find it out and try some of those, some of the multi-phase modeling on their own as well. And get back to you in case they need further help. Yeah. And the next session will start at 6:10 p.m., which happens to be 12:40 p.m. UK time. So we have roughly 20 minutes. No, yeah, 20 minutes of break. The link is provided by. Yeah, someone shared the link. Yeah, Shanu itself. Okay, yeah. So we will be staying online. All the participants can log off and rejoin again in 20 minutes time. Then we'll have the next session where we'll touch upon more case studies, exciting ones. So, Chandrakant, you may share the slide, the symposium slide, so that it can stay online. Yeah. Okay, welcome all again. Hi Nadine, yeah. So we are going to the next session. So that is, I'll just share the slide.
Did we say something?
Yeah.
Okay, it's good now. It's fine. I had a quick question for the next session. Do we plan to have 5 minutes or 10 minutes discussion in between talks?
5 minutes discussion.
Okay, good, thanks.
Yeah, so yeah, Tom is here, Thomas. So yeah, Nadine, you are chairing the session, so I request you to please introduce Tom and go and take it forward.
Okay. Yeah, first of all, welcome back everyone to this last session for today. It's my pleasure to introduce Thomas Williams. So he's been a PhD student at the University of Oxford, also working with Lee Sweetloff and George Ratcliffe. And he's been a lecturer in Brazil since 2013, if I'm correct. And he's currently working at the botany department at the University of Brasilia. So we're actually very curious to hear your presentation, and having this said, the stage is yours.
Yeah. Just I'm sharing.
Yeah. Okay, it's not.
Yeah, Tom, just a minute. Yeah.
Yeah, sure. No problem.
Yeah, there you go. Just passing the
Yeah,
presentation.
that's great. Just a second. Okay, hopefully that's appearing nicely. Let me just start the presentation. Okay. Okay, well, thank you very much, Nadine, for the introduction, and thank you very much also to George, to Nick, and to Shyam for inviting me to speak. It's really a privilege to be able to share a little bit of our research with you today. So during this presentation, I'm going to be talking about flux balance analysis of seed reserve mobilization. So the first thing we really need to think about then is why we should be interested in using flux balance analysis to look at this physiological process. Well, firstly, because reserve mobilization is obviously a part of a critical phase of plant development, during which the reserves that are laid down in the seed are remobilized and used to form the tissues of a photoautotrophic juvenile plant. And obviously this process is absolutely essential in the field for high yields. If we don't have efficient establishment of seedlings, then yield obviously goes out the window. It's also an interesting process because the reserves that are used for growth during this period are also often the products of agriculture, and we're talking about seed crops. So if we're interested, for example, in modifying the composition of seed reserves, for example, changing the amount of amino acid in storage protein, or maybe altering the degree of fatty acid in saturation to alter oil composition, or maybe even removing raffinose oligosaccharides from a seed, well, that could potentially have knock-on effects on germination and initial plant growth. Also, this is actually a rather curious metabolism that's going on. We detect large variations in composition between different species and between different seedling organs. So I'll show some data later, but I think everyone can cite an example of a seed that has a large amount of oil, or another seed that has a large amount of starch, or another that has a large amount of protein. And all of these different reserves have to be converted into seedling organs which have a relatively uniform composition. So there's a lot of interconversion of different biomolecules going on. And this interconversion obviously involves some interesting metabolism. So during this phase of growth, we see activated metabolic processes which aren't often seen in other phases of plant growth. For example, the glyoxylate cycle, gluconeogenesis, and amino acid catabolism are rarely seen outside of the metabolic what's the word? Metabolic sort of context, that's it, context of seed mobilization. Right, so during this talk, I plan to talk initially about flux balance analysis of soybean seedlings. This is work that we've been doing in collaboration with Maurice Cheung's group. So when I say we, a lot of it was done by Maurice, specifically the model building and curation, and we took a lot of the decisions about the modeling together. We'll be talking about our progress towards modeling of peanuts and peas. After that, I'll talk a little bit about how light influences reserve mobilization and how we may go forward with modeling that process as well. And finally, I want to talk about modeling reserve accumulation in soybeans in the field, which is a bit of work that we've started doing in the last year. So why soybean specifically? Well, soybean obviously has enormous commercial importance. Brazil produces more than 110 million tons of soybean per year. And in part, that's because of its chemical composition. The seed contains around 40% protein, about 20% oil, and a large amount of sugars as well. And there's a lot of interest also in the genetic manipulation of that seed reserve composition. As I mentioned, on modifying raffinose series oligosaccharides, modifying oil composition, modifying protein composition. And as I commented earlier, that could potentially affect the process of germination and initial growth. It's also a good model for experimental work because all of the reserves are essentially localized in the cotyledons. There's no endosperm to speak of in the developed seed, which makes it easy to work with, easy to dissect when we're talking about seedlings post-germination. And once we have good quality seed, we can also obtain rapid and uniform germination, which is a prerequisite for good germination and initial growth experiments. And finally, it's a species which has been studied extensively with regards to its genetics and its metabolism. So there's a lot of information available in soy psych and in soy base. There's also extensive compositional information available about, for example, cell wall and reserve proteins. So the first thing that we really had to consider when doing this work was the creation of a genome scale stoichiometric model for soybean. And so Maurice Cheung, who's going to be speaking a little bit later, built this model using the Soy Psych 6.0 database, which was the most up to date when we started doing this work. And the model he built and curated contains 3,000 reactions, a large proportion of which can be mapped to different soybean genes. There isn't all that much compartmentation information available for soybeans specifically, so we based the compartmentation on Arabidopsis thaliana. So the reactions are compartmented to the peroxisome, the mitochondria, the plastid, and the cytoplasm. And our model is capable of producing all of the known biomass components, or the biomass components that we regularly quantify through photoautotrophic and through heterotrophic growth. Once we'd done that, once we got our initial model built, then we had to consider how we would be modeling metabolism in seedlings themselves. So the first thing we did was to add reactions to describe reserve mobilization. So soy psych at the time was missing a number of reactions. My master student at the time, Thiago, did a fairly extensive search to look for the reactions that we needed to add. So those involved in degradation of RFOs, those involved in transport of fatty acids to the glyoxysome or peroxisome for degradation, amongst a number of others. We decided fairly early on that we were going to focus on early post-germinative heterotrophic growth, so that simplifies the system extensively. So we considered that we could model the seedling as essentially two compartments, one representing the sink, which would be the hypocotyl root axis, and one representing the source, which would be the cotyledons from which derive all of the reserves that are mobilized during the process of initial growth. So we duplicated our original model to reflect those two compartments, and we added reactions which represent transport from source to sink. So sucrose and amino acids, including GABA, which was a decision we took based on the information that's available in the literature. So once we got the model set up, then we had to populate the model with constraints on growth and development. So we carried out experiments germinating soybean seedlings in sand at 25 degrees in the dark. That's a system which produces uniform growth and allows rapid harvesting of the material. And we measured dry mass accumulation and reduction in the case of the cotyledons over time. So you can quite clearly see here that the dry mass of the cotyledon pair decreases over time. We can see here that the mass of the hypocotyl root axis increases, representing the transfer of the reserves from one organ to the other. There's extensive mobilization essentially from 24 hours onwards. So it was that period from 24 hours onwards that we decided to model because there's a significant change in the accumulation of biomass within the hypocotyl root axis. So what we did essentially, we opted to model in 3 different stages of development from 24 to 48 hours, 48 to 72 hours, and 72 hours to 96. Once we'd, of course, carried out the quantification of biomass, we needed to consider the composition of that biomass. So we carried out a fairly extensive analysis of the composition of biomass, quantifying protein from the nitrogen content, starch using an enzymatic method, organic acids and free amino acids using NMR, fatty acids using GCMS, and soluble sugars using HPLC. So we carried out a pretty comprehensive analysis of the composition of the material that we were harvesting, both the cotyledons and the hypocotyl root axis. And these analyses already show a degree of change in composition over time, which suggest exactly what metabolic processes are likely to be highlighted by the modeling process. So we can see, for example, the consumption of soluble sugars occurring. That's this red bar here. In the cotyledons, you can see that they're being degraded over time. The accumulation of starch. So soybeans basically have no starch in the seed, but the starch accumulates after the beginning of germination and during post-germinative growth. The the consumption of protein, which is clearly seen in the hypocotyl root axis, and liberation of amino acids, which presumably occurs to drive elongation of the hypocotyl root axis, and the consumption of fatty acids. It's important, obviously, to note, though, that this is all compounded by alterations in dry mass. So whilst in the cotyledons we don't see a decrease in abundance of protein, the protein is being mobilized because the mass of the cotyledons is decreasing over time. And we can use that product of the biomass abundance and the abundance of different biochemical components to calculate flux constraints for our modeling. So the next point, which is a critical point, which several people have already mentioned today, is the choice of an objective function. So in my previous experience with FBA was with cell cultures, where I've been involved in work where we would be able to increase the ATP maintenance cost effectively to match consumption of glucose or a single substrate to that which was determined experimentally. And that's not really very practical to apply in this situation because we've got multiple inputs into our metabolic network. So what we opted to do was to maximize ATP and NADPH maintenance consumption and then minimize total flux. And we found that that tended to give a good prediction of the amount of CO2 released by the model when compared to what we know is released by the seedlings, which we analyzed based on the carbon content of the seedlings over time. And of course, that doesn't really say much about the internal fluxes, but it does at least show that our model under those circumstances is pretty well mass balanced in terms of it's not accumulating spurious metabolites. There's another problem, though, when we start talking about multi-compartment models, which is how we can divide maintenance costs between 2 different organs. So to do that, we use the relative water content as a proxy for metabolic activity. So that's a little bit more difficult to support. So what we did to investigate whether that assumption was going to cause significant effects on our modeling outcomes was we modeled what the effect of alterations in this ratio would be on the distribution of flux, so the rate of a series of different reactions here in different ratios of cotyledon to hypocotyl root axis maintenance costs. And you can see that whilst there are quantitative differences, the qualitative conclusions that we can draw shouldn't be affected by that assumption. So what kind of results do we get out of our modeling? Well, firstly, we can see that a relatively small proportion of reactions are activated during all 3 phases which we considered, which is pretty normal for this kind of study because we only quantify a select few biomass components, which then constrains exactly which metabolite, which reactions are going to be activated. But what's interesting is that we can detect a switch in metabolism between the first period, so 24 to 48 hours, and the later two periods. So you can see here that whilst there's a core set of reactions, 409 reactions that's active in all three periods, the later two periods share fewer reactions with the 24 to 48 hour period than they do with each other. So it's 134 reactions that these two here share, and only 16 apart from the core set of reactions, and 20 here which are shared with the 24 to 48 hour period, suggesting there's a shift in metabolism going on. And indeed that's what's going on when we look at the fluxes in more detail. So these maps here represent the carbon fluxes through different reactions, here for the cotyledons and hypocotyl root axis for the first period. So what we can see here is that metabolism at this period is relatively low overall. The fluxes through metabolism are relatively small. There's consumption of soluble sugars in the cotyledons, which is fueling starch accumulation, and that soluble sugar is also sent to the hypocotyl root axis to support growth. Also in this organ, interestingly, citrate, which appears right at the beginning of germination and initial growth, is used to fuel function of the TCA cycle. In the hypocotyl root axis, here we've got the TCA cycle and glycolysis working as you might expect. But what's really interesting is what happens when we go to the next phase. So here we've got the cotyledons from 48 to 72 hours, and suddenly we see the activation of gluconeogenic metabolism. So we've got operation of the glyoxylate cycle here, which results in the production of oxaloacetate, which is then used to fuel the production of sugars, both starch in the cotyledons and also export of sugars to the hypocotyl root axis. You can also see here that the model predicts that other substrates can be used for gluconeogenic flux. So for example, glycine can be converted to glyoxylate and fed into the glyoxylate cycle. Alanine to pyruvate here also fed into gluconeogenesis via PPDK. You can also see here that during this phase of growth, fatty acids represent an extremely important respiratory substrate. So you can see that there's a cycle going on here that results in the transfer of reducing power from the peroxisome to the mitochondria. And that cycle is responsible for supplying about half of the NADH, which is oxidized by complex 1. And furthermore, all of the citrate that is produced here comes from fatty acids originally. So essentially, metabolism here is being fueled by fatty acid degradation. And so what we can see here in this graph here is that, as you might expect, from the initial period to the later two periods, transport of the phloem, of compounds in the phloem greatly accelerates. So we can see the loading of, or predict the loading of sucrose and different amino acids into the phloem, their transport from the source to the sink. And when we look at the sink tissue, which is the hypocotyl root axis, of course, we can see those different compounds being used. So in the hypocotyl root axis, there is de novo biosynthesis of amino acids and cell wall, which is necessary for growth and expansion of the tissue. But there's also a large amount of amino acids and sugars arriving from the cotyledons. Parts of those, some of those amino acids are used directly for protein synthesis, but others are fed into catabolism. So the electrons are passed directly to the electron transport chain, and the carbon fed in principally into the TCA cycle. So this is an interesting example where we can actually see or predict the use of amino acids as also as a respiratory fuel, specifically in the case of the hypocotyl root axis. So as a number of people have said already today, now these experiments that we do using flux balance analysis, they are predictions essentially. And we're interested eventually in carrying out 13C labeling experiments and performing flux quantification. And so this idea actually came from the fact that we carried out some previous labeling experiments which suggest that seedlings will reach isotopic steady state after about 4 or 5 days labeling. So my student Jansen carried out a series of labeling experiments using U13C glucose. And we could show that isotopic steady state is obtained between 4 and 5 days, and there is still growth of the tissue during this period, as can be shown here by the increase of the length of the hypocotyl root axis. So Jansen carried out extensive extraction of cotyledon hypocotyl root axis metabolites, fractionation, quantification of mass isotopomers using GCMS, and we're in the process of analyzing those data using 13C Flux 2, which is which is quite a challenge considering that we're having to simultaneously model the metabolism in 2 different subcellular compartments. So I'm looking forward to the talks tomorrow and hopefully find some insight about how we can improve the strategy that we're using there. So the other thing I wanted to talk about in this first chunk of the talk that we're doing in collaboration with Maurice is the effect of reserve composition on metabolism. So as I've commented right at the beginning of the talk, crops present significant diversity in seed reserve composition, and that even exists within botanical families. So within the Fabaceae, for example, you can see that pea, peanut, and soybean have pretty different reserve compositions. Soybean has a composition I already commented about, with a sort of large amount of protein, an intermediate amount of oil, no starch, whereas pea, for example, is almost the opposite. It has a large amount of starch and practically no oil. Peanut, here in the middle, has a decent amount of protein, huge amount of oil, and an intermediate amount of starch. So what we're interested in doing now is understanding how reserve composition might influence the metabolic processes that are activated during post-germinative growth, exploiting this biological variation, this natural variation that already exists. So to do that, we're essentially following the same pipeline that we did for the soybean work. We've quantified already the biomass composition of cotyledons and the epicotyl root axis for both of these other species. During post-germinative growth, we've measured biomass accumulation and removal from the cotyledons, and that's allowed us already to calculate input and output fluxes. And we're in the process of producing genome scale models using the annotated genomes of peanut and pea. And that's difficult because the pea genome is massive and the peanut genome is a polyploid. So that's quite a challenge, but that's something that Maurice and his team are working on at the moment. So we hope that in the future, we'll be able to generate genome scale models for these species and thereby compare flux predictions for these 3 species and hopefully better understand how the reserve composition influences in metabolism. So leaving this question of heterotrophic growth to one side, what comes next? What happens if we want to explore metabolism a bit later on in seedling development? Well, going back to soybean cotyledons remain attached to the plant for quite a long time, several weeks typically, and they're green. They have significant photosynthetic activity, at least enough to offset respiratory carbon loss. So that means if we want to go further, if we want to model slightly older plants, then that means greater complexity. It means that we have to understand how light influences photosynthesis of the seedling and plant development. So a student of mine, Karen, carried out a series of experiments using young but green soybean seedlings, looking at how different light intensities can affect reserve mobilization, specifically looking at what happens during a day-night cycle and what happens under different light regimes. So we grew plants to the cotyledon stage, which is more or less what you can see here. The cotyledons are open, the first true leaves are in process, are expanding. And we then subjected them to different light regimes, so low light or darkness, and then we analyzed the biochemical composition of the different organs. So this heat map here summarizes what we saw when we compare plants under standard light conditions to plants kept in the dark for 3 days before analysis. And as you might expect, we see some of the effects which are common to carbon starvation in, for example, Arabidopsis, which is probably the system that's been most investigated. So you can see depletion of carbohydrates in both the leaves and the roots. But interestingly, we also see the accumulation of osmolytes, including amino acids and certain sugars in the epicotyl. And that's again likely to be necessary to fuel osmotic expansion of the cells that's going on as the seedling etiolates under the darkness conditions. As I mentioned, we were also interested in looking at the change in metabolite abundance's biomass composition over time, right? So what we've got here is the analysis of biomass that was collected at 6 in the morning, so the beginning of the light period, at 6 in the evening, so the end of the light period, and 6 in the morning of the following day. And we did that to get a handle on which metabolites are actually capable of accumulating and then being degraded during the night. And so we can see from this that, as you might expect, starch is accumulated during the day, quite clearly here in leaves, and then degraded during the night. And that cycle is abolished in our darkness treatment, which are these dark gray bars here. And this analysis also uncovered an oscillation of asparagine, which occurs over a diurnal cycle in the epicotyl. So this study was done partly because we wanted to look at more how light influences reserve metabolism in soybean, but also to collect data that we might then be able to use for modeling. So we plan now to construct a multi-compartment model of soybean, similar to that for the heterotrophic seedings, but obviously expanded to include more compartments. We plan to constrain that model using measurements of biomass composition and of growth. Try to model metabolism over a day-night cycle, which will use the same sort of concepts that Maurice came up with the dial modeling, which other people have already talked about today. And then we want to try and predict the effects of light intensity on metabolism and try and compare that with our experimental data. And hopefully this will then feed into other experiments that we plan to do in the future, where we're going to be trying to model photoautotrophic soybean plants. And in that regard, I wanted to finish talking very briefly about what we're trying to do in the field. So we've been looking at reserve mobilization, but we're also interested in the flip side of the coin, which would be the production of those reserves, which are later going to be mobilized. And this has been investigated using MFA. Soybean embryos was subject to extensive study, principally by Doug Allen's group, where they showed, for example, the influence of light on refixation of CO2. But of course, the question remains, what about implant, because these are isolated embryos, and what about under field conditions? And a particular interest we have is in how water restriction may affect reserve production. Brasilia is a particularly dry part of Brazil. We have more or less 4 or 5 months per year where it doesn't rain at all. And we also have, during the rainy season, we may have what are called veranicos, which are 2-week periods of very little rain and very high temperature. So we're interested in finding out how water restriction may affect reserve production. So what we're thinking of doing is using flux balance analysis combined with a control field experiment to predict metabolic fluxes during reserve synthesis. How's that going to work? Well, we're interested specifically in this phase of soybean development, so R5 to R6, which is the period of grain filling. So roughly at around R5.1, the plant is more or less mature in terms of its aerial parts and in terms of its root system, and it's beginning the accumulation of reserves within the embryos. So we're looking at more or less this phase of R5.1 to R6.1 over a more or less a month-long period under 4 different levels of irrigation. And you can see here the experimental system. We've been very fortunate to piggyback on a lovely experimental setup in Embrapa Cerradas, which is just down the road from the University of Brasilia. They've got a very clever system where they grow plants during the off-season, where there's no rain at all, and they can control exactly how much water is applied to the crop. And you can see here a gradient of height of these soybean plants going from well-watered to very little water on the right-hand side here. And this here is showing more or less the stage of development at which we begin our analysis. So we've already carried out the biomass analysis. We went to the field, we took plants out, and we've quantified the biomass invested in leaves, the pod, the stem, and the embryos. And we can see that this experiment is going to capture certain physiological, certain developmental processes that we know occur during grain filling in soybeans. So you can see here this large decrease in embryo mass per plant as a function of decreased water availability. So these are the 2 most watered situations, and these are the 2 least watered situations here. So there's this large decrease. What's interesting, though, is you can see here that this blue and the green bar are essentially the same. These are not the same treatment. There's a decrease in water availability for the green treatment. But so the embryo mass is the same, but the way in which that embryo mass was produced must be different, because if we look at this graph here, you can see that the leaf mass is preserved for this high water treatment, but it is not for this lower water treatment here. So this remobilization of reserves present in the leaves is presumably associated with the capacity of the plant to maintain investment in embryos. So obviously we're collecting this data, we've got our biomass data, we're carrying out a biochemical characterization. What are we going to do in terms of modeling? Well, what we want to try and do is to use, again, a multi-compartment model to try and predict metabolic fluxes during this phase of growth. So we have also, thanks to our collaboration with Embrapa, we've got a lot of meteorological data. So we've got daily light, temperature, and rainfall. And we can combine that with a photosynthesis canopy model for soybean with physiological measurements of maximum photosynthesis, light response curves, specifically for in leaf dry mass, to predict gross carbon fixation in field conditions. So that will then be pumped into our leaf compartment of the model, and then the leaf will distribute carbon to these other subcompartments of our model. And obviously, again, we'll be looking at using dial modeling to reflect what happens during a day-night cycle. And again, we'll be combining this with our analysis of biomass composition. This is obviously work that we're going to be doing quite a bit further down the road, but I thought it was interesting to share today so I can get some input from the experts in the field. So just to conclude what I've been talking about today, firstly, that fluxes in soybean vary extensively during post-germinative growth, with a large switch in metabolism towards lipid and protein catabolism at around 48 hours post-imbibation of the seed. Stoichiometric modeling will eventually allow us to investigate this relationship between reserve composition and seedling growth, and may allow us to make predictions about how metabolic engineering may affect capacity of a seed to germinate and to grow. We've shown that light has this important effect on reserve mobilization in soybean, and we hope to incorporate that kind of data also into our understanding of seedling establishment. And finally, we aim to extend this multi-organ approach, which we started with the original soybean model, to soybean plants in the field and test the effect of water availability on metabolism during grain filling. And so finally, I'd just like to thank the people that were involved. Firstly, Maurice, we've had this long-term and excellent collaboration going on with his group at NUS Yale. Thanks also to Walter Quadras at Embrapa Cerrados and Maria Lucrecia Ramos from the UNB, who we've got this excellent collaboration with for the field experiments, the University of Brasilia NMR facility for analysis, and the team that's been involved in all of these analyses. So Christina, Vincent, and Umberto Nasa are working on the field experiments. Thiago and Lucas did a lot of the work for the soybean seedling metabolism and the other two legume crops as well. Janderson and Victoria did the labeling experiments, which we're in process of analyzing. And finally, Karen did a large chunk, did pretty much all of the work on light with some help from Bruno with regards to the lipid quantification. And thanks also to our funders, CNPq, FAPDF, and the University of Brasilia. And thank you very much for your attention.
Okay, thanks, Thomas. It was an excellent talk, and also thank you very much for staying in time. Before we start the question session, can I quickly ask the organizers, do we have 5 or 10 minutes for questions now? So we're starting at 20 past, right?
Sorry. Yeah, we have about 9 minutes, Nadine. Yes, please go ahead.
Okay, so the first question, Thomas, can you elaborate more on how light intensities affect protein starch reserve?
Yeah, sure. So what we found was that, so we got 2 different light treatments, a low light and a darkness treatment that were applied after, we analyzed the effect of it after 3 days of application of the treatment. And generally both of those led to reduction in starch abundance. The effect on protein is less clear. So what we proposed in the article that we published on this was that the soybeans seem to prioritize an accelerated mobilization of reserves that are not protein. So it seems to prioritize accelerated degradation of lipids and of starch when we put cotyledons or when we put the seedling in the dark, but not actually the protein content. That seems to be relatively preserved. The reason for that, I'm not sure. Perhaps because nitrogen is a relatively more precious reserve to a plant than the carbon reserves themselves. But yeah, that would require further investigation to know exactly why.
Okay. Ed wants to know, do you think incorporating Sanu's FBA approach into your soybean model would change the flux distributions? And how much of the initial growth is cell division versus expansion?
Yeah, so that's a really good question, and unfortunately, the grow model came out after we'd already, well, perhaps not unfortunately because we might have had to change everything, but yeah, I think there would have been some big differences. So what happens in seedling growth is that, yeah, it's mainly elongation. There's not a lot of cell division going on in the hypocotyl root axis. Is basically elongation. So actually, it would be a perfect test case for this that kind of analysis. Of course, what happens, because we carried out this pretty extensive characterization of the biomass composition, a lot of the osmolytes are already included in our modeling. So they're there, but they're not predictions in the same way as the GROW model would make. They're not derived from equations, they're quantified. So our approach has the advantage that we know it reflects what's actually happening in vivo, but the disadvantage that it's less predictive than the GROW model. So I think actually there's a decent chance they might give the same results, but of course, as Sanu mentioned before, the advantage of what he's doing is that you can try and understand why it happens, whereas what we're doing shows what is happening.
Okay, thanks. So the next question, how do you identify which metabolites are transported between time tissues?
Yeah, so that's a good question as well. Initially, what we tried to do was to look at the phloem composition. So there are protocols published for doing that. Unfortunately, when we did it, we actually spent quite a long time using those constraints for our modeling. But we quickly realized that there were spurious results there. So the phloem composition contained large amounts of hexoses, and we know that in soybeans those aren't transported in the phloem. So we had to ditch that data in the end. And so what we then decided to do was to basically allow as much freedom as possible to the model. So we allowed it to transport all of the 20 amino acids plus GABA and sucrose, because the evidence suggests that sucrose is basically the carbohydrate that's transported in soybean. I didn't really get into this, but the model does a pretty good job of predicting what is transported in the phloem, but it's not perfect. So we see a lot of transport of glutamate, whereas in reality it would probably be more glutamine and more asparagine. So there are some things that it gets right and other things which it doesn't. But that's one case actually where we left the model with quite a lot of freedom to decide what it should be doing.
Okay, any more questions from the audience? Maybe while we're waiting for more questions, I can ask you one or two questions. Okay, let's take this one first. Did the light intensity affect LEA protein?
I can't answer that question, unfortunately, because we don't quantify proteins on that kind of detail. But well, yeah, sorry, I can't say. I would imagine so, because they're so abundant in soybean seed, but yeah, I can't answer that one.
Okay, so maybe I can ask my question. You pointed out earlier that in your model, your objective function was to maximize ATP and NADPH, and your system is a really tricky system. I mean, it doesn't really have an obvious objective, so as compared to looking at a growing leaf or even a mature leaf. So did you actually test other objective functions, and did that have an effect on your model solutions?
Yeah, so we did. And initially, we'd thought about just minimizing total flux, but what happened when we did that was that the model tended to it would not produce any kind of maintenance metabolism at all, because that increases the sum of the total fluxes, and it tended also to accumulate peculiar metabolites. So it would release gases that we know that the plant doesn't release, or accumulate secondary metabolites. And so obviously what we could have done is to constrain all of those fluxes to zero, but we really kind of rethought what we were doing at that point. And we sort of, our rationale was that, well, if we're measuring everything that's going in and everything that's going out, the carbon that's left over is really most likely to be released as CO2. And if it's being released as CO2, it's probably because it's the product of maintenance respirations. That was our logic there. And also Maurice had already demonstrated that that two-part ATP then minimization objective function, at least for Arabidopsis cell cultures, that does a pretty good job of predicting accurate fluxes in vivo. So that's why we went with that one.
Okay, there's another question from the audience. Are those measurements free amino acid pools which might act as potential carbon and nitrogen sources?
In terms of the seedlings in the light, yes, they are. So what we do is that at the moment we estimate protein using the nitrogen content, using combustion to determine nitrogen content. We estimate protein from that. We measure the amino acids using the most abundant amino acids using NMR. And then we subtract that nitrogen, the nitrogen invested in free amino acids, from the total nitrogen, so that we don't double count the nitrogen that's in protein. So yes, potentially those amino acids are a store of carbon, and the model predicts for the soybean seedlings at least that under certain circumstances the amino acids are degraded. So there's degradation of glutamate, there's degradation of the branch chain amino acids, there's degradation of lysine predicted by the model. More or less using the routes that the pathways that have been sort of studied for Arabidopsis.
Okay, maybe I can have, okay, okay, another question. Amazing science, good work, chasing the problem from every angle. Have you looked at changes in leaf biomass composition during senescence?
Oh, yeah. So that's what we're going to be doing, actually. So if we go here, yeah, you can see that. So the leaves from the C2 to C3, which is more or less R5.5 to R6.1, depending on the water treatment, senescence is happening or not. So in the orange and red treatment and the green treatment, there's senescence going on. So we've actually measured leaf chlorophyll over from those two time periods to have a sort of physiological idea of what's going on. We measured gas exchange, so we know that photosynthesis is decreasing over that period, and we're going to be measuring composition of the leaf matter, and yeah, there's definitely senescence going on. So we're quantifying protein and lipids and all those kind of things, and I expect actually we'll see some of the metabolic processes that we saw in the seedling in terms of the glyoxylate cycle again being activated in our model during that process.
Okay, great. Thanks, Thomas. I think it's time to move on to the next speaker.
Okay, hopefully that should have.
Yeah, just
So our next speaker in today's session will be Maurice Cheung, and you've heard his name already a couple of times. I'm just waiting for the slide to pop up.
Yeah, there you go.
Yeah, so Maurice also obtained his PhD at the University of Oxford, and he then straight moved on to being an assistant professor at the Yale-NUS College in Singapore. And without really giving a lengthy introduction, I really want to give you the time to talk about your research. So I will hand over to you, and we're really looking forward to your presentation, Maurice.
Hey, thanks, Nadine, for the introduction. And while I'm waiting for access to share my slides, so hopefully everyone can hear me. So if not, you can use the chat function to let me know. So throughout this talk, feel free to use the chat function and type everything, because I'm quite used to looking at chats while, I guess, well, in my teaching. So feel free. Okay, I think I can share. Okay. Right. Oh, interesting that I actually don't get to see the chat, but that's fine. Okay, so hopefully everyone can see my slides. Okay. If not, then the
Yes, Maurice.
yeah.
Go
Okay.
ahead.
Okay. Good. Good. All right. Then I will start. So first of all, thanks, George, Nick, and Shyam for organizing this event so that all of us can get together virtually. And also, of course, for the invitation for me to speak to you here today. And I also want to add Lee in the mix to thank all of them for their guidance and mentoring throughout my PhD days and beyond as well. And of course, I want to thank Tom, who was actually the one who got me started with this guard cell project. Yeah, so, and also, of course, for presenting our work just now on soybean metabolism. And finally, I want to thank my undergraduate student who did basically most of the work, so George Rathan. So he's done picture on the right. So he did most of the simulation work in the modeling and also wrote the draft of the paper. Okay, so a bit of an introduction of more like undergraduate research, really. So I'm currently working in Yale-NUS College, which is a small liberal arts college in Singapore. So it's more of like a US style small liberal arts college. And it's a collaboration between Yale in the US and NUS, National University of Singapore. One thing that I just want to highlight is that it's a primarily undergraduate institution, meaning we don't have any graduate students or no masters and PhDs. So I've, well, for the past 6 years, been tailoring my research towards working with undergraduate students. So here I just want to share a bit about my experience with working with undergrads, and I see it doing undergraduate research as more of an education as well as like science. So just as an example, Joshua joined my group actually when he was a first-year freshman student, and I spent about 2 years working on this guard cell project. One thing I just want to say is that in Yale-NUS, students only get to choose the major in the second year, and Joshua actually ended up being a philosophy major, which students can choose whatever they want, and like they investigate their interests. So how I see doing undergrad research is really to allow students to explore their interest and really as an education for teaching undergraduate about the process of doing science and allowing them the opportunity to do science. So I would encourage you all to also give it a go and do more undergraduate research. Okay, all right, that's enough for talking about undergrad research. So let's go into the science. And to be fair, I'm not sure how many of you are actually cares that much about guard cell research, but so I think what my talk will be, like the narrative of my talk is more on sharing my experience on how to do like an FBA kind of study in the field of plant metabolism. So the journey starts, of course, by picking an interesting metabolic system to start with. Okay, so in my case, now I see this guard cell project is more like a case study to demonstrate this process. So okay, in this case, guard cells, which I think already mentioned a few times throughout the symposium. So they control the opening and closing of stomata, so the air hole in the leaves. And how they do it is by accumulating osmolytes. So thanks, Sanu, for introducing that. So I don't need to tell people what osmolytes are. So osmolytes accumulates in the guard cells when the stomata opens. Right? So when the osmolyte accumulates, of course, the water comes into the guard cells as well, and it forms a particular shape that then allows air to, CO2 to enter the stomata, but also water to get out. Okay, and of course, when the stomata closes, then the osmolyte will get either degraded or exported, so that then water will also get exported. So the common osmolytes for guard cells are sucrose, potassium, malate, and chloride. So those are the main ones that people have observed. So for my work here is only on C3 plants. So in C3 plants, what happens is that the leaves, the stomata open during the day and they close at night. So that's the general idea of how these stomata opening and closing works for C3 plants. And one thing that's interesting that I didn't know before I get into this project is that different osmolytes actually accumulate at different time of the day, so different phases as well. And so you can see that there are 4 phases, which you probably can see from the title of my talk as well. Okay, so after we've picked an interesting system to study, then the next step is really to think about what are the open questions that people are really asking in this field, right? And I must say, for me, before Tom talked to me about guard cells, I didn't know that much about it. I know what they are, but I don't know about the metabolism, exactly what are the open questions. So the thing we need to do is to, of course, read into the literature and find out more about what are the questions people are asking. So in the case of guard cells, there are quite a number of things that we still don't really quite know what their roles are. So for example, like why certain osmolytes accumulates at different times, or is this even just like a choice of that? Why are there so many different osmolytes? Why don't they just use one particular one? Right. So there's sucrose, there's potassium, and assuming that the guard cell accumulates potassium, which of course is a positive ion, right? Then of course you can't just accumulate positive stuff. Then the guard cell will become very positive. So it has to balance out with some negative charges. And the counter ion for potassium could be malate, or could be chloride, could be a combination of both. Again, there's a question of why, which one, right, and why. And then the third one, which I think is the most interesting one from the metabolic, Flux's point of view, is the synthesis and catabolism of malate. So assuming that the guard cell accumulates malate, then how is it being either synthesized or degraded, right? So this is you'll see later, basically. Okay, and then the other two questions are basically the roles of sucrose, external sucrose in this case, because guard cells also take in sucrose from the surrounding mesophyll cells. So then the question is, what are the roles of the sucrose? Is it mostly just to provide the osmolyte to be accumulated, or could it also have other energetic, like provide as a energy source for guard cell metabolism? And finally, the interesting one is also starch as well, because guard cells do accumulate starch, but not exactly the same as mesophyll cells, meaning they actually have a different pattern in starch accumulation over the Yale cycle. Okay, right. So now that we've have a interesting system, and then we have a list of questions to ask, then of course the next thing is to think about how to answer those questions. And first, you need to identify the tools to do it, really. Okay, and in this case, because I work on metabolic modeling, so of course it's going to be metabolic models. And in this case, I just want to highlight the importance of having a mass and charge balance model, which I think Sanu and Nadine has already mentioned. So this is a case study on why I would say mass and charge balance models are important for doing FBA modeling, at least in certain use cases, right? One is, well, actually, before I go into it, I just want to say that, so George and I, and together with Kemi, actually spent quite a lot of time to make this, mass and charge balance model a core model of Arabidopsis. And in this case, we've actually added metabolites or defined some metabolites with multiple charges or charge states, but that's just something extra. But in general, having a mass and charge balance model allows us to really study the balancing of charges and I guess in particular protons for things like chem plants, where chem plants accumulates a lot of malate, and of course as well. Actually, I should say malic acid, right, because it's quite acidic when large amount of malate is stored. And yeah, so in that case, then the balancing of protons could be quite important in terms of the energetics, because there's a lot of energy used for just pumping protons. So in my case, because I'm studying also osmolyte storage or accumulation, and that also includes malate as well as other charged ions, right? And so we will need to have, I will need to use a mass and charge balance model. Okay, here you can see a few minor changes that I actually made to the model in terms of how we model protons compared to the previous versions of it. So this is more in the technical detail. But basically, one thing that I did was to block the so-called proton leak from the cytosol, and that also allows certain leakage reaction. One is from the extracellular space to the cytosol, and the other is to allow protons to so-called leak from the vacuole to the cytosol. So of course, these are all unidirectional, meaning of course, on if you want to do it the other way around, you have to pump, you know, to use energy to pump protons in the other direction, but allow protons to leak from that direction. And the other thing is that I allowed free exchanges of protons between the cytosol and the mitochondrial intermembrane space, which, to be fair, it looks like it could make a big impact, but in the end it didn't. So I just left it at that. And finally, one other things that I've changed is to allow protons to accumulate in the vacuole, which I think was a bit different from how Sanu or our previous version of the model, where we fixed the or we tied the proton accumulation with the malate or citrate accumulation. Okay, so those are the small changes that I made. Okay, right. I think you've seen this diagram a number of times, so I don't think I'll spend too much time on this, other than to say that whenever you're tackling, a new problem, there could be cases where you need to develop new tools to study the new system, right? You've already seen all the two-phase models. I won't go into it. But I'll just show you this four-phase model, which is, I guess, the extension of the two-phase model. Of course, is a lot less complex than the ones that Nadine and Sanu has presented. It's just four phases. But I guess my philosophy in general for modeling is that models should just be as simple, as complex as it needs to be, meaning as simple as possible to the extent that you can model a system that you're interested in. So that's my approach. So in this case, for guard cells, well, I needed four phases, and that's because in the morning, actually I should start at night, because at night the stomata is closed, right? So that means there's low osmolyte level in the guard cell. But then when it comes to the morning, stomata opens up, right? And that's the time when the osmolyte starts to accumulate inside the guard cells. And then throughout the day, I just assume that the stomata stays open, and so it will just have high osmolyte content. Of course, in actual leaves, depending on conditions, they might open and close the stomata multiple times, and yeah, but that just means that the opening closing process happens many times. But in my case, I just want to keep it simple, so I just assume that cyst stays open. And of course, then in the evening, then the stomata closes, then that means the stomata close, so the osmolyte will either get degraded or exported. So those are the four different phases, which has, I assume there's four different steady state basically throughout the 24-hour cycle. Okay, so as you can see, these arrows there, right, unidirectional metabolite accumulation reactions, which some people call it linker reactions. And I guess one thing to note is that in this case, the phases are actually has different lengths, and that can cause some complications in terms of how to interpret the fluxes. But first of all, I want to say that, okay, so for each phase, which you've seen before, right, we need to set phase-specific constraints such that they will actually have different behavior. So in this case, of course, during the morning and day, there will be light, right? Whereas at night there will be no light. The other thing is, which I think all speakers has highlighted, is the which I'm quite happy to hear that everyone highlighted the fact that maintenance cost is an important part of, especially for plants, where plants spends a lot of energy for maintenance. So I did include a maintenance cost here as well. And in this case, right, it's scaled by the hours, right? So because of the fact that each phase has different lengths, that means, the maintenance reaction for, say, the morning, will actually, no, I'll say the one for the daytime, which is 11 hours in my model, right, will be 11 times larger than the one in the morning, which is only 1 hour. So you have to scale that accordingly. So then once you run a simulation, get the results, right, then you'll get the fluxes. And one thing to note, if you've tried doing this kind of a phase or multiple phase with different lengths, and then you'll see that the results you get are per phase, right? So meaning you get what happens, the total amount of flux for the whole 11 hours for the day and the night. So of course, if you want to make comparisons, depending on what comparisons you want to make, you might need to normalize it to per hour. So that's one thing to note. The other thing to note is that, okay, metabolic accumulation. So this might not be a very intuitive concept for I guess, people who have not done it before. So basically, if you look at this graph on the bottom right, so I think you can just focus on this one here on the one basically which is starch with the circle in the middle, right? So this is actually how starch level changes for guard cells, or at least for some guard cells. Again, guard cells are actually very flexible in the sense that in some guard cells, different guard cells might do things differently depending on conditions, depending on species, and so on. But in this case, you can see that the starch actually get used quite a lot in the morning, right, and then start to accumulate again during the day. So getting back to the point of metabolic accumulation. So the size of these arrows, right, on the left diagram basically corresponds to, think of it as the metabolic level at the end of that particular phase, or like the transition of the phases. So that's how you should interpret metabolic accumulation in this kind of model. Okay, then next I'll move on to challenges in using FBA. So this is more of like a caution for those who are new to FBA. So first of all is that FBA often we use efficiency-based objective function, which actually means that the FBA, the most efficient route is usually chosen by the FBA algorithm in the Flux solution, and not any other alternative routes that are slightly less efficient. So which could be quite a big problem with we want to explore alternative pathways. So that's one of the things you need to realize is that when, for example, in guard cells, something I want to test is say which osmolyte is the most efficient. Like the use of different osmolytes, right? And how that affects metabolism. Of course, if you just run FBA, it will just choose the most efficient one, and that's it. So what you need to do in order to actually model the use of other osmolytes is then to constraint, set specific constraints so that you can explore the use of other osmolytes. Okay, so my philosophy of FBA, I think, is similar to Tom and others, right? Is that I would want to allow as much flexibility as possible, so that then you let the FBA algorithm to choose the preferred routes. But okay, so one of the ways in which you can, I guess, go around the problem of exploring alternative routes is actually to do something called flux sampling. I did a bit of that during PhD, but there are many other algorithms which can do flux sampling that could allow you to explore other alternative routes, but I'm not going to go into that today. Okay, all right, then I want to talk about the power of FBA, or why we actually do FBA in the first place. And the main thing that I think many people have already talked about is that it's a predictive modeling method, meaning it actually gives prediction, which of course has its pros and cons. Okay, so of course the pros is that you actually don't need that much data in terms of the input, and it can give you a large amount of predictions. But of course, the downside is that you have to be very careful when you interpret the results, because these are only predictions. So that's something to keep in mind. Okay, maybe I'll just share a bit about the specific constraint that I use for this model. So actually, there are just really 3 constraints. One is osmolyte levels, right? So basically, how much osmolytes accumulates at the end of each phase. And then the other thing is that we know that some sucrose is being being imported from the outside. So in this case, I said 80% based on some literature value, but of course, there's actually a wide range of literature value. In this case, you just have to stick with one. But then one thing that you can do is then to explore the effects of changing this parameter on the rest of the fluxes, which I'll talk about later. And the rest, and then the final one is cell maintenance, which I'm not going to talk about, but it's an important part. And the objective function in this case is to minimize just the amount of photon, so energy, really. And with something called parsimonious FBA, so PFBA, so that just basically means minimizing the total sum of fluxes as a secondary objective function, which I would encourage all of you to do, and because PFBA does have a lot of advantages in terms of reducing the amount of, or actually just eliminating substrate cycles and so on. Yeah. Okay, so and then of course, as a FluxMAPS talk, I will have to show a FluxMAP. People enjoy looking at FluxMAPs, so but I'm not going to go into any details except for to say that actually, you can see that the framework can predict fluxes for all 4 phases and also the metabolite or osmolar accumulation. Okay, so here is a slide that's, I would say, more educational in a way for people who might be new for FBA. And of course, like the usual constraint that we set is that, okay, right, how much flux can go through this particular reactions. But as Sanu already mentioned, there are actually other type of more complex constraints that you can set. One is sum of fluxes constraint, meaning you can say, all right, reaction 1, flux of reaction 1 plus reaction 2 plus reaction 3 equals a certain amount, right? So in this case, or in my case, I actually use a bit of that where I say, okay, right, the total net charge of osmolyte being accumulated has to be 0. Right? So that's a constraint. Then in which case, then you can say the amount of potassium being accumulated minus the amount of chloride because it's -1 charge, and then minus 2 times the amount of malate because it's the 2 minus, right? And that should equal to 0, right? So you can set a constraint like that, and that means if the cell has to accumulate a potassium, it has to take in at least some chloride or malate. Similarly, there's another constraint on just the sum of osmolytes, and in this case, I have set a particular number for amount of osmolyte, that total osmolyte that's used that needs to be accumulated to open the stomata. Right? So in this case, similarly, is like the total amount of osmolyte has to add to a particular value. Okay, so that's sum of fluxes. Then the next one is the flux ratio constraint, which is more of actually very similar concept. In this case, is to say, okay, so flux of reaction 1 has to be maybe like 3 times of flux of reaction 2. Okay, but if you rearrange the formulas, basically you can do it the same way. So we use that quite a lot when we talk about Rubisco, carboxylase, oxygenase ratio, or 3:1 ratio. But in this case, it's also quite useful for setting the constraint for the different lengths of phases as well. Okay, now I think I should move faster because we're getting out of time soon. So, okay, this is something I want to highlight is the thing that I want to highlight is what George has already highlighted, which is that FBA in general, or even PFBA, only give you one of the many possible optimal solutions, right? And so one thing to note is really that if you see something that happens in PFBA, don't do it. I'll say get excited about it, good, but always check your findings with something called flux variability analysis, so FVA. And in this case, it's actually a very good example to show why it's important. So, I ran a simulation, and then one thing that I was interested in is actually, how melee was being degraded. And, I've looked at the PFVA solution, and it says, oh, okay, so maleic enzyme was the only one that was predicted. So good, right? So I got the answer. But then if you look at the FVA results, which actually gives you the possible range of fluxes based on the primary objective function, then actually see that both the maleic enzyme and the PEP-CK gives you actually a large flux range, meaning it can actually use both. Yeah, so in this case, you should double check your flux prediction with FVA, and that's, I think, a very good example to show why we should do FVA. Okay, and then here is an illustration of how you actually set constraints to model a particular scenario. So basically, people do a lot for C4 and CAM, where there are different subtypes, and you want to measure model different subtypes. And the way to do it, or at least one of the ways to do it, is really just to block the other alternative routes, right? So you force the model to only use a particular osmolyte, say in my case, right? So for example, I want to look at the use of malate versus chloride, and so what I do is, if I want to model the use of malate, then what I do is I block the chloride accumulation and vice versa. So in this case, actually surprisingly, that we see that the malate and chloride, the use of malate and chloride is actually quite similar in terms of the energetic efficiency. And the other interesting part, as you can see, is that the starch accumulation pattern is also different between the two. Okay, and then final 5 minutes, I'll just go to talk about scans, which is a technique that you can use for looking at the effects of changing one particular parameter on your flux prediction. So in this case, I tried to look at sucrose, how sucrose intake from outside changes the flux prediction. And in this case, obviously, you take in more carbon, sucrose, right? Then you need less photosynthesis, and ultimately, after you've got to a point where you have too much sucrose, then you might start to degrade your sucrose as energy source, and then start to accumulate starch. And okay, so this one, right, was the second last slide, which is about, I guess sometimes the joy of doing FBA really is like finding surprises. So in this case is finding an alternative route for like the Kelvin-Benson cycle, where on the top is the diagram that you've probably seen many times, right? So it's a traditional one, where there are two dephosphorylation steps, right? So that's the FPBAs and the SPBAs here, right? So there are two steps. But then actually, when I looked into the actual fluxes, and then I saw that in, if I remember correctly, it could be the open phase, right? Where I see that it doesn't use these traditional, I guess, enzymes that are in the Kelvin-Benson cycle, but instead it uses PFP to actually make pyrophosphate. And of course, investigate further. Okay, then why is it producing pyrophosphate? And that's because actually, when the guard cell is accumulating osmolyte, right, it actually requires energy to pump protons, and pyrophosphate can be used as an energy source for pumping protons. So in this case, it actually saves a bit of energy by producing pyrophosphate through these dephosphorylation steps. Okay, so I would like to end by, I guess, sharing with you a bit about my experience of being I see myself as a theoretical computational biologist, meaning I really, do more theoretical work and more basic science. Yeah, and so how I go about basically doing this kind of work is that you first of all need to understand the strengths and weaknesses, or like limitations of your modeling technique. So in my case, FBA, there are things that's good for, but of course there are limitations. You can't do certain things with FBA. So having that knowledge, then you can go and find interesting biological questions that your modeling technique that right, can be used to answer. Of course, to do that, there are many ways. You can read papers.
Maurice,
Yep.
sorry to interrupt you, but for the sake of giving the audience the chance to ask some questions, can I maybe ask you to
Wrap up very quickly. Okay.
Wrap up. Thanks.
Sure. All right. I'll just wrap that. Right. So basically, don't be afraid to develop new techniques if it's needed for your modeling, and don't be afraid to dive and learn about new problems. And there'll be a lot of learning. It'll be difficult, but it'll be fun. So enjoy the modeling process. Okay. Thank you.
Okay, thank you very much, Maurice. I really didn't want to cut you off, but I think there are some interesting questions from the audience that you want to answer. So maybe we dive into the discussion.
Sure.
There was one question: when studying flux improvements after some gene deletion, is it better to compare the minimum flux or the maximum flux of wild type and mutant?
What do you mean by minimum, maximum flux? Yeah, that's not quite sure what you meant by that. But there are multiple approaches to study mutants. Yeah, there'll be MoMA, there are Room, there are many algorithms that we can talk about.
Yeah, so maybe we'll move on with the question, and the person who asked the question can specify in the meantime. Another question was, you mentioned about secondary objective function. Does this mean that within multi-objective functions, you can sequence them based on priority or rational decisions?
Yeah, so in this case, it's like you apply the first objective function first, you get the solution and get the objective value, then you set the objective value of the primary objective function as a constraint, and then you do the second part, second step. So it really depends on what your system and what you want to model. Right? The choice of objective function is the difficult part for FBA, I would say. So it's I'm happy to talk more, but it really depends on the system.
Okay, the next question. Does the model suggest anything about the use of ATP generated in the chloroplast for the Calvin cycle versus other processes, such as osmolyte accumulation?
Interesting question. I didn't look at like the proportion of how much ATP is from photosynthesis versus like I guess breakdown of other things. But I must say for guard cells, it really depends on how much sucrose is taken in from the outside, and I don't think there's a good answer to it because different guard cells could be different.
Okay, let's have one more question. How was the new metabolic route in the Calvin cycle discovered?
Well, to be fair, okay, let's put it that. That's a prediction that we make, and I must say it's like then down to experimentalists to go and verify and whether actually see if PFP is active in the opening phase of the guard cells. And how is it discovered? Is really just you run a simulation, you look at the fluxes, and this is actually the tedious part where you actually have to look into the large amounts of flux predictions and find the details.
Okay. For the sake of time, I'm skipping the remaining questions, but I hope there will be some more time to discuss this later.
Thank you. I'll go in the chat and answer those. So.
Yeah, thanks again, Maurice, for this very interesting talk, and thanks for highlighting some crucial modeling assumptions that are really important also for the students in the audience. So thanks again.
Welcome. Thank you. To work out how to stop sharing, which is this. Okay.
Yeah. Jyotika, you can flash on the next speaker, please. If there is a problem, you can transfer back to me. I can share. Okay, that's good.
Okay, so let's move on to our next speaker.
Yeah.
This is Chandrakant Joshi, and he is actually a homie because he's a PhD student at the IIT in Mandi, working in Shyam's group, and he'll be giving a more theoretical method-driven presentation. So the stage is yours, and we're looking forward to your presentation.
Thank you so much, Dr. Nadine. So, yeah, Jyotika,
Share it to CK. Yeah.
yeah, if you, Jyotika, if you go on the top, there is a small red strip. Then you have to click that, stop sharing, and then you can transfer the rights. Thank you, Jyotika. Yes, so hello everyone. I'm Chandrakant Joshi. I am I am a PhD scholar here in IIT Mandi, and I'm here to talk about efficient system-wide pathway comparisons in multiple organisms for defining precise metabolic features. So as we have all been attending all the talks today of the flux balance analysis in the plants, and we will attend some more talks regarding the 13C in tomorrow as well. So I think we all understand how important it is to have to understand the metabolism and to have the precise metabolic networks properly defined in an organism. So to start with a very simple example, of let's say three fruits, which are all red: strawberry, tomato, and apple. Oversimplified example, and they are all red. And our goal is to see what is causing the color to appear in a fruit. So we may look into its genome to see what is happening in the genome, and see the metabolic features, and the same and this subsequently we can check the genome of strawberry as well as apple, and we can manually identify what are the similarities in the systems and what are the differences. So uh, so um, so now building on the similar concept, my question is, if we have the genome information, can we actually overlay that genomic information on the metabolic networks, and and see what is happening at the metabolism level, and mainly identify the metabolic capabilities of different organisms. So if you go in the NCBI, there are like thousands of genomes in all the categories available, sequenced and annotated. And if we open the KEGG, we have like thousands of bacteria and few hundreds of eukaryotes and archaeas. So based on these, we what we can do is we can either compare the metabolism at the gene level, or we can compare them as the gene function level. So my work focuses on the gene functions and how the gene functions are defined, and then once we have the gene functions, how we can backtrack that information to with the genome, and find out the metabolic capabilities. To briefly explain the orthologs, the KO identifiers, which is KEGG orthologs, they can identify help us identify the gene functions. So I don't work with the FBA or on the but I do work with the metabolic networks. So I work in the adjacent FB adjacent area. So if we all know the orthologs, paralogs, and homologs. So for example, the early globulin genes for the globulin. So it may have been diversified in a speciation event, and then alpha chain and beta chain was maybe we got two different chains, and then now we have alpha chains in frog, alpha chain in human, and maybe mouse. So these three genes, they may have different sequences, and they may have the sequence similarity may be reduced, but ultimately they retain the same function. So based on the function, orthologs are defined where we have the same functions. So in the KEGG, if a particular gene is doing same function, even if the sequence has been changed, if it is doing the same function in a metabolic network, it is defined as a KEGG ortholog, and then it is assigned a KO identifiers. Then the KO identifiers, now the KO identifier across the different organisms can be compared to actually see the contrast in their metabolic capabilities. So like KEGG allow us to reconstruct pathway based on the KEGG mapper tool. So here I use KEGG mapper reconstruct pathway. So here you can reconstruct the metabolic pathways of up to 10 organisms. So for example, I have taken 3 genes in organism 1 and 3 genes in organism 2. You can see here in the box. And then if I execute, it will try to overlay those genes, the genes on the metabolic pathways based on their KO number. So these K 00844. So for example, this is one KO number, and you can see in this like very small examples, 2 KO are the same and 1 KO is different. So based on this, I can like execute, and then in the 2 organism, something like this I can generate from KEGG. So I can get the pathway information. The green shows it's color coded, so we can see the capabilities of both the organism. But in reality, when we are working on the genome level, there are thousands of genes involved, and if we talk about multiple organisms, maximum 10 in the KEGG's case, then we have tens of thousands of genes and their KO identifiers. So in that case, we needed an automated tool to get the KO number. And if you see here, it's the numbers. The file is in a particular format. So we needed something to get the KO numbers from the KEGG. As I found out that there was no straightforward way to get the KO numbers from the KEGG. And the KO numbers are available, but there is a lot of unnecessary data. Not unnecessary, but lot of extra data there. So to overcome this limitation, I designed a web server. So this is the third iteration of web server, my web server called G2Q. So first was the command line based. The second where you had to download some file. The second iteration was also command line based, but the tool would work in the background. And third is the web server based, which can be accessed by this link. So what this web server does is, this is designed by our lab, by me, and it is hosted at IIT Mandi. So what this web server does is, I'll explain in detail, but basically if you want to compare the KO numbers of multiple organisms, you just write the name of organisms and their codes here. In the background, it will send the request to KEGG. It will download the data. It works in 2 steps. So it will retrieve the data, process the data in the particular formats needed, and then a text file is generated. And now you can it will prompt you to download this text file in the downloads. And then in the KEGG pathway reconstruction, you can simply upload this file without any editing. So this file will be in the format required by the KEGG pathway reconstruction tool, and it will contain all the genome level genes, tens of thousands of genes and their KO identifiers. So then these genes can be annotated against the pathways in the KEGG, and then we can visualize the pathway in the KEGG and color code it, and then based on that, based on the comparison, we can gain biological insights. So yeah, this work was published last year in the interdisciplinary sciences, computational life sciences. So to give a brief summary of the web server can efficiently help compare the metabolic networks of multiple microbes. It's a very simple web server. If you don't have any leads to go on, you can simply click here, and it will prompt you to download a text file again, which is downloaded from the KEGG in real time, and it will have all the genomes which are available at that time in the KEGG and their code. So you can see 3-letter codes here and the organism name. So and eukaryotes as well as bacteria, and then based on the availability, we can select the organism of interest. And then there is also instructions on the web server, and then it will ask you how many organisms you want to compare. In theory, you can go up to thousands, but for the visualization, second step of visualization, the KEGG only supports up to 10 organisms. So let's take an example of 3 organisms, and then if you click next, it will in real time it will generate 3 boxes. You can put the name of the organism. So these are 3 organisms I work with. So I for example, I put their name, Thermobifida fusca, and its code. And then if I click on the download KO identifier, it will download a text file as I suggested. The single text file will have all the 3 organisms KO numbers, and then this file can be uploaded by clicking here in the KEGG Mapper Reconstruct tool. So we can either copy paste the content of the text file here in the text box, or we can simply upload the file. If we click on the execute, it will show a list of all the pathways in the KEGG where at least one gene has been mapped. So in our case, we can see 440 pathway were found where our gene of interest were mapped, and then let's say let's check what's happening at the glycolysis level. That's the simplest pathway. So in the glycolysis, if you see, we can see the contrast. The boxes are color coded now. The white means absence of that particular gene in the first organism or second organism, depending on where the white space is. So let's say my goal is to find out the lactate production, which organism is capable of producing lactate. The question can be different based on your need. So for example, you can have what kind of sugars your organism can metabolize, or what kind of different genes your organism can break down, what kind of different secondary metabolites your plant can make. So based on that, I can see clearly here that only third organism, which is Geobacillus thermoglucosidaceous, has the ability to produce lactate from pyruvate. Other 2 organisms, they don't have, they are missing the gene to produce lactate. So this is one very small example of how the G2KO can help in quickly contrasting the metabolic capabilities of organism based on what capabilities we are looking for. So now using the G2KO web server, I have done 4 case studies. The first is I'm going to present 3 case study, but yeah, the first is the comparing the amino acid biosynthetic pathway in selected organism. We know the 13C, especially the 13C experiments, they rely heavily on the retrobiosynthetically mapping the metabolism. So we isolate the amino acid mainly, and then based on their labels, we decipher the label of the central carbon metabolism. So I did a comparative study to see the amino acid biosynthetic metabolism. The second was the central carbon metabolism of the phytopathogen Ralston solaniferum. The third case study is a small case study to showcase that G2K can be adopted to work with plants as well. So comparison of a few selected fruits and their secondary metabolism to see if their capabilities to produce some nutritionally relevant metabolites. And then fourth study is the designing of the synthetic microbial consortia towards the targeted application. So I designed based on the GTKO in my initial PhD work. I designed two synthetic microbial consortia. Now I have validated them and experimentally validated them and optimized the bioprocessing, which I will not be discussing that today, but I will focus on the first three case studies. So first case study is the amino acid biosynthetic pathways using the G2KO, and what insights can we gain? So amino acids are the labeling in the amino acid is very important. This is what we measure actually in the 13C experiments. So as you see in the cell, when glucose goes in, and ultimately if the glucose is labeled, the label will end up in all the different amino acids across the central metabolism. And then if we can measure the labeling pattern and the extent of labeling in one particular amino acid, we can decipher the fluxes of that particular node in the metabolism. So few interesting takeaways from this study I found. So if you see the M9, if you focus on the M9, which is the Pyrococcus horikoshi, so here you will find a lot of biosynthetic pathways are missing. So I was actually I was also I was surprised to find an organism which has lost almost all of the ability to produce the amino acids. But then I checked again. I thought maybe if there is an issue with the annotation or something. But in the literature, I found out that this is an obligate heterotroph. So in order to grow this organism, we have to provide almost all the amino acid with the media, because this organism cannot biosynthesize any of the most of the amino acids. Then second interesting difference which I found in my study is that M1 and M2, both the Geobacillus, Costophilus and Parageobacillus thermoglucosidases, they were missing the R3 gene of the serine biosynthesis. So they can't really make serine from this node. But in all the 13C studies, which have also been done in our group, in our lab as well, we have found out the serine is always labeled. So the traditional route which we consider for serine, that is from 3-PG to serine, which is not possible in the Geobacillus. It's not possible in the Geobacillus. So, but still, we find the labeled serine. So there must be some other homologue working there or some other alternate pathway. And lysine metabolism is of another interest. So lysine is being metabolized by two different routes. And they are both from, one is from aspartate and another is from AKG. You can see the number 27, 29, and 30. And number 27, if we compare here, it is predominant only used by the bacteria, the route 27 from aspartate to lysine. On the other hand, from AKG to lysine is utilized by fungus and archaea. But the fungus and archaea, they also split their ways in between. So the pathway 30, reaction number 31 is utilized by the fungus, while the 29 is utilized by the archaea. So these are few of the insights we found on the amino acid biosynthetic pathways. Then in the Ralston solaniferum, I did the central carbon metabolism comparison of the phytopathogen plant phytopathogen Ralston solaniferum, and I find out that it has got the ability to produce the PHP. So it's a phytopathogen, but it has got the ability to make bioplastic, which is interesting because it can be adopted. It can be grown on the glucose, which is relatively cheaper, and it can make bioplastic. Similarly, in the central carbon metabolism of the Ralston and Solani serum, we find out that in the central carbon metabolism, in the glycolysis, key regulatory genes are missing. So fructose 6-phosphate to fructose biphosphate, it is not possible because the gene is absent in all the Ralstonia strains. We find out. So this work is experimentally validated by Poonam from our group. She'll be presenting this work tomorrow in detail. So there you can see how we got the lead from the G2KO, and then from that lead, how did we expanded, how did we hypothesize, and then worked by 13C labeling. We validated that actually it's utilizing the ED pathway to continue this metabolism. The glycolysis indeed is absent in the Ralstonia. So this work was published last year in NEM Systems. Um, then to showcase the potential of G2KO in the plant metabolism, I compared the secondary metabolism of few selected fruits. The table you can see on the right, just to see what kind of insights we can get. So, for example, this one part of the pathway, the narangin, which is secondary metabolite, narangin is present in only in the orange and the grape. So they are the only two fruit which can biosynthesize this metabolite, and some other metabolites of nutritional importance, like polyphenols, like like here we can see that this one, so all the organisms are able to biosynthesize the polyphenol, but except the wine grapes, the cucumber and the tomato. So all the other fruit has got so, this metabolite. So like this, we can, based on our question, based on the query we have, we can expand the capabilities of G2KO to check the capabilities of plants or any other eukaryotes as well. So to summarize the case studies, the G2KO can be adopted to retrieve a huge number of organisms at a time. Although there is a limit in the KEGG of the visualization, that you can only visualize 10 at a time. But if you're downloading that huge number of data, thousands of genome, in theory you can download. If you're downloading that data to curate either your genome scale metabolic reconstruction, or to aid in your 13C, defining your network for the 13C, it can be done in a go. You can download a lot of organism and their gene information. The second is G2KO is very rapid. If you want to compare, let's say 10 organism, it will only take minutes to accentuate the differences and similarities in the metabolism. So you can you can actually see what kind of capabilities one organism has. There are a few limitations of this tool as well. The tool depends. So currently, it is for annotation. It is depending on the KO identifiers. So it depends on the KO to annotate annotate the metabolic networks. And again, the limitation to reiterate, if you want to visualize, only 10 organisms can be visualized at a time. So yeah, I would like to thank IIT Mandi, my PhD supervisor, Dr. Sham, and all my MedSys Biolab members for continuously supporting this tool. They have been also using this tool, and they're always helping their hand in the testing the tool and reporting if they find any bugs. Thank you.
Thank you very much, Chandrakant. Are there any questions? You touched upon quite a lot of topics in your talks. That was really impressive. Thanks.
Thank you.
So there was one question from the audience. Does KEGG reconstruct pathway predict subcellular compartment? And then you partially answered this in your talk, but maybe you still want to comment on this?
Yeah, so KEGG, currently KEGG doesn't account for any subcellular compartments. Some of the very specific metabolites or pathways, if there are any, they are accounted for. For example, some of the pathways in the which are very specific to the mitochondria, or some which are specific to the chloroplast, they are separated. But if there is a common pool in between the cytoplasm and mitochondria, they are not separated, so they are not differentiated in the KEGG.
Are there more questions? Yeah. The lack of subcellular localization, could that be the reason why SR labeling didn't agree with your map?
Okay, so yeah, that's interesting. Interesting question. So yeah, the lack of subcellular localization maybe, but Geobacillus is it's a bacteria. So in the bacteria, it's I'm not. I mean, there are. I'm sure there is some separation, but we at the time of modeling we don't consider any separation. So I think it might be some homolog or some other enzyme which is acting as the acting on the to biosynthesize the serine. But yeah, the similar observation was made in the 2018 paper in some other group. They were working on the 13C mapping, and they also found the same in some other geobacillus. So yeah, that's interesting thing to explore.
Okay, are there more questions from the audience? Okay, so then I've got a question. So if I wanted to use your tool, are there any specific requirements regarding the installation, or is there anything, or do I need to have certain coding skills, or?
So yeah, this version is a web server, so you can simply visit, and this will not prompt to download anything except the data which you are requesting from the KEGG. So it will only download that. There is also an offline version. I didn't share the link for that one, but if you want, I can provide an offline version. So that is that there everything is in the command line. But yeah, at the user end, user don't have to download anything other than the scripts. And then the command line is also not very user friendly, but it will step by step ask you what to do. So it will ask, okay, enter the name of first organism and the second and third. But it can get it if you have, like, if you want to download thousands of organisms, that can get a bit hectic if you, because in command line you can't simply paste it. But in the web server, you have the ability to copy paste if you have a single in a single file.
Okay, thanks. There's another question from the audience. Can we view the pathway of drug metabolism in clinical MDR strains of pathogens?
Okay, so drug metabolism, I'm not sure about the drug metabolism, but the KEGG does have nice curated pathway of the genobiotic degradation. And there I did find some pathways for the okay, he's asking drug metabolism in clinical MDR strains of pathogens. So I'm not sure, because the drug metabolism, I think it may get a bit complex. There are so many things involved. But yeah, if the pathway, either if the pathway is present in the KEGG, then definitely you can overlay your KO data based on the tool, and you can overlay your KO data on the pathway.
Okay, are there more? Okay, Here we go. There's the link. Any more questions from the audience? A technical one. Did you have to get a license, academic from CAC, to download huge data sets?
So yeah,
To comment on.
yeah, not the license one, because what I'm basically downloading is mostly text. So it's a bit tricky. CAG doesn't specify how much we can download, but for the academic, they just instruct that keep it simple. So I'm using CAG's own API in my programming, that is CAG's REST API. So yeah, CAG definitely is aware that there is a tool which is using their API to talk with them. But yeah, it's not licensed. I don't have academic or commercial license.
Okay, thank you very much. Are there any further questions? Okay, I don't see any burning questions at the moment, and I think we're right in time to move on to our next speaker, and this will be the last speaker of the session today.
Yeah, Jyotika, please flash the speaker slide, please.
So I'm the host now. I'll share the slide in a minute.
Okay.
Yeah, so moving on to our last speaker, this will be George DiCenzo, and he's currently an assistant professor at the Department of Biology at the Queen's University in Canada. And yeah, as I just said, he'll be giving the final talk of this session today, and we're very much looking forward to hearing about plant-microbe interaction, I think. So the stage is yours, and we're looking forward to your presentation.
Thank you. So I'll just wait until I'm able to share my screen. But while waiting, thank you for that introduction, and thank you for the opportunity to speak today. I'm looking forward to talk about this work with you all. There we go. Great. So, yeah, as was mentioned, I'll be presenting some of our work looking at the integrated metabolism of a legume and its bacterial symbionts during a nitrogen fixation in a legume nodule organ. So as a bit of background of why we're interested in this topic, nitrogen is a very important macronutrient for plant growth. In agricultural systems, it's often a plant growth limiting nutrient. And so people apply nitrogen fertilizers to their crops or even in their yards. So this graph just demonstrates the significance of nitrogen fertilizer to our society. So this x-axis is time, the y-axis is human population. Shown in gray is the estimated proportion of the human population that would be supported without synthetic nitrogen fertilizers. Well, the part in red shows the percentage or the proportion of the human population who are thought to be alive today as a direct result of synthetic nitrogen fertilizers increasing crop yield. So you can see about 3 billion people today are alive dependent on nitrogen fertilizers. So these are obviously very significant and important contribution to human society, but they do come with drawbacks. So the synthesis of nitrogen fertilizer involves the use of the Haber-Bosch process. So this is a process that uses 1 to 2% of the global energy supply each year, thus creating large amounts of carbon dioxide and other greenhouse gases. And then runoff from agricultural farms can lead to eutrophication of water bodies and other environmental hazards. So this, for example, is a algal bloom in the Gulf of Mexico, caused in part by runoffs from agricultural fields that are contaminated with excess nutrients like nitrogen and phosphorus. So what we want to be able to keep up these crop yields, we need to find a sustainable way that reduces the impact of agriculture on our environment. One way to do that is to make more use or improve our use of rhizobium legume symbioses. So rhizobia are soil bacteria that live completely happy by themselves in the soil, but they're also able to enter into an endosymbiotic relationship with legume plants. During this relationship, the rhizobia take atmospheric N2 gas and they convert it into ammonia that is provided to the plant as a source of nitrogen. This allows the plant to grow and thrive even in nitrogen deficient soils. So to illustrate the benefit of the symbiosis, I like to show these few pictures here, where half of the plants are inoculated with rhizobia and the other half are non-inoculated. And so I think it's pretty clear to see that the inoculated plants are much healthier than the non-inoculated plants, just demonstrating the potential of these rhizobia to increase plant growth even in the absence of nitrogen fertilizer. So in some cases, but not always, rhizobial inoculation of agricultural fields can fulfill most, if not all, of the nitrogen needs of the plant. So to look a bit more at the establishment of the symbiosis, this is a little schematic showing the initiation of the symbiosis and what the nodule organ looks like that contains the rhizobia. So that begins with a exchange of signals between the plant and bacterial partners, which leads to the bacteria infecting the plant through an infection thread, and then the bacteria released intracellularly within plant cells. These infected cells eventually form this nodule, and the nodules, like those of plants from the Metacago genus, which is what I'll focus on later, the nodules have spatially distinct developmental zones. So these different zones are labeled here. And zone 1 is the apical meristem, so the nodule keeps growing and it's not infected with bacteria. Zone 2 is the infection and differentiation zone. So this is where bacteria are released into plant cells and the bacteria undergo a process of conversion into a state where they can fix nitrogen. This interzone is a small layer of cells that its function isn't completely understood. And then zone 3 is where the nitrogen fixation actually occurs. Zone 4, which I won't focus on later, is the senescence zone. So this is where things die. So yeah, so later on in our modeling, we will represent the different zones of the nodule. And so it's good to keep in mind that zone 1 is uninfected, zone 2 is where differentiation is happening, and zone 3 is where nitrogen fixation is actually occurring. To see what this actually looks like, and not just with the schematic, the top left image is an image of an alfalfa root nodule. So these are on the scale of millimeters in size, so they're not microscopic. They are visible with by eye. So if, for those of you that garden or work with plants or soybean, for example, you may see these structures on the roots. So they have this characteristic pink color due to the presence of leg hemoglobin that helps maintain a low concentration of free oxygen, as the nitrogenase enzyme that fixes nitrogen is oxygen sensitive. It's not very evident in this image, but the tips of the nodules are white. And so those white parts correspond to the zone 1 and zone 2, where the nitrogen fixation is not yet happening. This is a transmission electron micrograph of part of a nodule. And all I want to really show from this is that this here is the outline of one plant cell. And each of these dark structures in the cytoplasm are individual nitrogen fixing bacteria. So you can see that the plant cells are really chock full of these bacteroids. As a final point, on the right are 2 fluorescence micrographs of a rhizobium, known as Sanu Rhizobium malodi, the model system we'll work with in this presentation. And so both panels are the exact same strain of Sanu Rhizobium malodi. The difference is that these top ones were grown in a test tube. These bottom ones were purified from a legume nodule. And you can clearly see that the morphology of these are very different. So the bacteroids are much larger. They're polyploid, having up to 20 to 24 copies of their genome. They also have massive changes in the transcriptome, proteome, etc. So it's a really dramatic change from being a free-living bacterium to a nitrogen-fixing one. So the overall objectives of my research are kind of twofold. So the first part is to characterize the metabolic properties of rhizobia, as well as the metabolic interactions that occur during nitrogen fixation that really allow this process to happen. The second part is to get a better understanding of the function of rhizobial genomes and prokaryotic genomes more generally. But in most bacteria, about 30% or so of genes are genes of unknown function. And so one thing I want to do is to better understand what these genes are doing. And that really feeds back into this first objective, in that if we don't understand what a third of the genes are doing, how can we properly understand the metabolism of the organism and the metabolism important for this symbiotic nitrogen fixation? So we do this using a variety of approaches, both experimental and dry lab. And for today's talk, I'm going to focus on work that I did mostly as a postdoc, finished off in my current position, but mostly done during my postdoc studies with Alessio Mangoni and Marco Fondi at the University of Florence, in which we use metabolic modeling to look at the metabolism of the inoculated legume. So if we want to look at the metabolism of the inoculated legume and the interconnected metabolism of the two partners, the first thing we need is a rhizobial metabolic model and a legume metabolic model. So the main model system I work with is Sanu Shameer, and so we worked on building a metabolic model for this organism. So this was done through a series of stages. Our first version of the model was published in 2016, our most recent one in 2020. So it's something we worked on over 5 years or so. And I won't go into too much detail so I can focus on the integrated model, but the final model contains 1,348 genes and about 1,400 reactions. To prepare the model, we incorporated a large number of different information. So we referenced 240 literature sources throughout the model. So one advantage of working with SMLOD is there's a large amount of literature on its metabolism that really helped us build a high quality model. We generated biologic growth data for mutant or wild type and mutant strains of SMLOD to better understand what carbon sources it can grow on and the different genomic regions involved in these carbon sources. And we incorporated what is known as transposon sequencing data. So this is a functional genomics method that lets you identify essential genes for growth and environment on a genome-wide scale. So we were able to use this method to identify all of the essential genes in SMLOD and use that to refine the model and ensure that the model predictions are in line with the experimental data. So I won't spend much time on this, but just to show that this is a reasonable quality model. This Venn diagram is showing predictions and essential gene predictions. So this red graph, or this red sphere, are the genes that were predicted as essential in their manually produced core model. So this was built by hand. And so that's kind of what we expect are the true core or essential genes. This blue one is our most recent version of the model, or core version of the model produced through integration of T&seq data with the program T and core. And this green one is our original version of the model, with also a core model of it. So you can see that there's much better overlap between our new version of the model and the manually produced core model than there is between our original model and the core model, confirming that a new model is a much better representation of the essential core metabolism of SMLOD. We also have high accuracy and growth phenotype predictions. So overall, we were on a different 162 carbon sources, we tested growth. Overall, we have 91% accuracy between the predictions of the model and experimental data, which is higher than the 84% we saw in the original model. In particular, we dropped the false negatives in half, from 18 to 9. So overall, this SMLOD model is what we think is a pretty good or strong representation of the metabolism of this organism. So that's the first part. Then we also need a model of a legume that's a symbiotic partner of SMLOD. So I'm a microbiologist by training. I didn't really want to get into building a complex plant model, but fortunately for us, that had been done by someone else. So this was done by Thomas Fowler et al., and they described a metabolic model for the organism Metacago, which is a symbiotic partner of SMLOD. So the first thing we did was we updated the model based on the newer version of the Metacago genome. So that just made the model more consistent with the most recent genomic information for this organism. And so this final model is a compartmentalized model containing various different organelles, and it contains about 2,500 genes and 2,900 reactions. And in the work from Thomas Fowler et al., they prepared scripts to make it tissue specific. So scripts to make a representation of shoot metabolism and a representation of root metabolism. So with these two models in hand, we set out to create our integrated model. So we did that in a series of steps. So we started by taking the metacago model and using the existing scripts, split it into a shoot model and a root model, allowing for nutrient exchange between the tissues as well as with the external environment. We then took the original metacago model, we took our cynorhizobium model, and we placed the cynorhizobium model within the metacago model, allowing for the exchange of nutrients from the cytoplasm of metacago to the cytoplasm of cynorhizobium and vice versa. So I mentioned metacago nodules have multiple different zones, and so we wanted to represent this in our model. And so we made a representation of zone 1, which is just the original metacago model at this point. And then we duplicated or made 4 versions of our integrated model to represent the distal and proximal zone 2, the interzone, and the nitrogen fixing zone 3. These different nodule zones were then connected to the root. So we allowed for exchange of nutrients from transfer of nutrients from the root to each of these different nodule zones, as well as the export of some nutrients from the nodule to the external environment. And the only nutrients that were allowed to go back from the nodule to the root were from zone 3, and those were asparagine and glutamine, which are the nitrogen export products of the nodule. So the only thing that could go back to the root from the nodule was the fixed nitrogen. So up to this point, the 4 different versions of the zones of the nodule were all identical. That's not very accurate. We know that the transcriptional and proteomic profiles of the different zones differ. And to try and better represent this, we integrated RNA sequencing data from this Ruedel study published in 2014, in which we used laser capture microdissection to divide the nodule into each of these 5 zones, and then used RNA sequencing to determine the transcriptional patterns of both the plant and bacterial partner. So we did this using Jimmy in the Tiger toolbox. And I won't get into the details of the algorithm, but essentially there's a somewhat arbitrary threshold of what expression is considered on versus off. And so genes with an expression above that threshold are turned on, those with the expression below that threshold are turned off. Then there's an algorithm to turn back on genes that are essential to maintain growth. And so in this way, we end up with slightly different metabolic networks in each of these 5 different nodule zones. And so just as a reminder, the zone 1 corresponds to the uninfected zone. These 3 middle zones correspond to the differentiation zone, and this last zone is nitrogen fixation. So one thing we did do with this model, and something that we could explore more, was to look at how metabolism differs in each of the zones, and look at how gene essentiality differs, so which genes are essential in which zone. I won't go over that data today, and instead I'll focus more on the bigger picture analyses that we did. So first, just as a quick summary of the model, the final model contained a total of 750 or so SML-OD genes and 1,300 unique metacago-truncatula genes. The reason I say unique is because several of these genes occur in multiple different zones. And so in this number, I've only counted them once, even if they've occurred in 7 different zones. So we do see distinct patterns of metabolism and gene essentiality in the various zones, although I won't go through that today. And one thing that was nice, and from what I understand is consistent with the literature, is that about 30% of the carbon fixed by the plant is sent through the nodules to help support the nitrogen fixation. So kind of highlighting the significant investment by the plant to have nitrogen fixation occur, as it is an energy intensive process. So if we compare some of our predictions to published data, published growth rates for Metacago sativa, which is a relative of Metacago truncatula, are about 0.1 gram per day per gram dry weight, which is about twice what we get in our predictions. So our predictions are reasonably close to the experimental data, although not exact. Similarly, experimental data show about 7 microgram per hour per gram dry weight of nitrogen fixation of Metacago sativa plants, whereas we get a value of 3. So again, it's a bit lower, although what is nice is the ratio 3:7 is almost identical to the ratio in the plant growth rate. So we have this consistent level of under prediction. And then the carbon cost for nitrogen fixation. So the gram of carbon needed per gram of fixed nitrogen varies in the literature, but is generally between 3 and 5 gram of carbon per gram of nitrogen. Our model predicts at 4.2, so we're right within this range, giving us confidence that our model is a reasonable estimate of the metabolism occurring in a nodulated lagoon. So we started with a somewhat simple analysis to look at the cost of symbiotic nitrogen fixation. So in this graph, the x-axis is the amount of ammonia that's provided in the soil or that the plant can use, and the y-axis is the growth rate. So nitrogen fixation is an energy-intensive process, and if nitrogen is available in the soil, the plants will use that before relying on nitrogen fixation. And so you can see that as we give the plants more ammonia, you get faster rates of plant growth as they rely less on nitrogen fixation. And so I don't show it, but you would see decreasing rates of nitrogen fixation if I plotted that data. So in this particular set of data, we allowed the rate of nitrogen fixation to vary, but we forced the plant to still have a nodule, even if it wasn't fixing nitrogen. We repeated the analysis, but instead of keeping the nodule and changing the rate of nitrogen fixation, we set the rate of nitrogen fixation per gram of nodule to be consistent and allowed the rate of nodule the gram of nodule per gram of plants to decrease. And so we see the same thing where you have increasing growth rate as soil ammonia increases, but you get higher growth rates because now the plant's no longer supporting that nodule tissue. So based on these analyses, we can estimate that the cost to growth, or plant growth, for symbiotic nitrogen fixation is about 28%. So the plants are about 28% slower when relying on nitrogen fixation than when provided ammonia. That can be further split down to 33% of that cost being due to the cost of maintaining the nodule, and 67% of the cost due to the actual process of nitrogen fixation. So we then wanted to do a somewhat more complex simulation that may better represent real life situations. So instead of just varying either nitrogen fixation rate or varying the rate of nodulation, we wanted to vary both. So we wanted to see how changing the efficiency of nitrogen fixation impacts plant growth. So along this x-axis, we have nitrogen fixation efficiency, which is the rate of nitrogen fixation per hour per gram of dry weight. And the y-axis is the plant biomass. And so when we did these simulations, we set this nitrogen fixation efficiency to a desired value. And then we optimized the amount of nodule biomass that maximized overall plant biomass. So the ratio of, say, shoot tissue to nodule tissue could vary across these different simulations. So as just to emphasize that, at this end here, 2% of the plant would be nodule biomass. At the far extreme, 26% of the plant is nodule biomass. So I mean, that's not really realistic. I just imagine these plants with these giant nodules. But for this initial simulation, we allowed that to happen. So what we see, what we see with this analysis is that there's diminishing returns to increasing the nitrogen fixation efficiency. Initially, you get this nice steep curve, and then eventually further increases to nitrogen fixation efficiency have reducing improvements on plant growth. So the half maximal rate of plant growth was achieved with just 10% the maximum rate of nitrogen fixation. And I wrote down the numbers so I don't forget. Yeah, if you decrease the nitrogen fixation to 50% of the maximum value, you only decrease plant growth rate by 10%. Now, as I mentioned, it's not really realistic to have 26% of the plant being nodule. It does vary based on the efficiency of nitrogen fixation, but I don't think any plants in nature will be 26% nodule. So we repeated the analysis, but limited the nodule to being either 10% of the total biomass or 5% of the total biomass. And so again, we see overall the same sort of pattern, but where you have this diminishing returns, but it's slightly different between them. But overall, the same pattern of diminishing returns remain. So this was interesting to us, both from an evolutionary perspective of whether it's better to have lots of, from both the plant side or the bacterial side, if it's better to have lots of nodule with low efficiency or high, small amounts of nodule with high efficiency rhizobia. And the benefits, I mean, the plant would probably prefer one, well, the bacteria will have, their preference will depend probably on the genetic diversity in the soil. But it's also interesting from biotech perspective of what do you want to focus on when improving nitrogen fixation? Is it more important to increase the amount of nodules that's formed, the efficiency of nitrogen fixation? And it was also a nice I guess, confirmation of what a number of people in the community are saying about engineering nitrogen fixation into cereals, where we'd expect that if eventually successful, initially this would be a very poor symbiosis, but even a very poor symbiosis is likely to have a noticeable impact on plant growth. So it is worth pursuing those efforts. So I'll go through one last simulation we did, and this was probably what I found to be the most interesting simulation we ran with this model. So we know that the rhizobia that are fixing nitrogen in nodules are provided C4 dicarboxylates, so malate, succinate, and fumarate, predominantly malate, as the carbon source that they then use to fix nitrogen. However, in our initial simulations with the model, the bacteria are being given sucrose, not C4 dicarboxylates, as the carbon source. And so that's opposite of what we see. We know that's not the case in real plants, and so we wanted to understand why the model wanted to give sucrose and not C4 dicarboxylates. So we tried to force the model to use or give the bacteria C4 dicarboxylates instead of sucrose, and that just caused the model to fail. It wasn't possible to use C4 dicarboxylates by the bacteria. We found out that this was due to a need of proton transfer between the plant cytoplasm and the bacterial cytoplasm. So initially, we did not allow the transfer of protons from one organism to the other, as we wanted to keep this the energy of the two organisms separate. So we didn't want, say, the electron transport chain of the bacteria to be powered by protons from the plant. But we do know from experimental work that the area around bacteroids is acidic. So you maybe can see on the screen, maybe not. Around these, around the dark bacteroids, there's this thin membrane you can see that encompasses them. So that space between the membrane and the bacteroid is acidic, and that's mainly a result of the import of protons from the plant cytosol. And so the only way we were able to get the bacteria to use C4 dicarboxylates was if we allowed the bacteria to use protons that were pumped into the zone by the plant, suggesting that there may be proton transfer between the plant and bacteria, which was a new discovery or a new prediction. However, that alone was still not enough to get it to use, or the model to predict C4 dicarboxylates as the preferred carbon source. So if we force the model to use C4 dicarboxylates, the plant growth rate was about 6% slower than if the bacteria were allowed to use sucrose. And so we thought maybe this might be related to oxygen. So we set an overall limit on the rate of oxygen usage by the nodule, but allowed the FBA to determine where the oxygen was used. Is it used by the bacteria or the plant tissue? But we know that, or estimates suggest that there's about 50 nanomolar of free oxygen in the nodule. And experimental data show that the Km towards oxygen of the bacterial terminal oxidase is about 7 nanomolar, which is well below the concentration of oxygen, whereas that of the mitochondrial terminal oxidase is between 50 and 100 nanomolar Km towards oxygen. So given those numbers, it's reasonable to hypothesize that the bacterial terminal oxidase may not be oxygen limited, while the mitochondrial terminal oxidase of the plant is oxygen limited. And so we changed the simulations, and instead of setting an overall limit on oxygen, we allowed the bacteria to have as much oxygen as it needs, and instead limited the activity of the mitochondrial terminal oxidase, which is dependent on oxygen. And so the x-axis here is the rate of mitochondrial terminal oxidase activity. The y is the rate of plant growth. And so not surprisingly, if you limit the mitochondrial terminal oxidase in the nitrogen fixation zone of the nodule, you get a decrease in plant growth because you're making less ATP. What was more interesting was the other fluxes that we can see. So we can ignore the purple one for here, but the key ones to look at are the red and blue. As you limit the mitochondrial terminal oxidase activity, the amount of sucrose being provided to the bacteroids decreases, the amount of C4 dicarboxylates provided to the bacteroid increase. And at this point here, you get a complete switch from using sucrose by the bacteria to using C4 dicarboxylates as the preferred carbon source. So this suggests to us that perhaps one of the reasons that nitrogen-fixing bacteria use C4 dicarboxylates as a carbon source is due to this low oxygen environment in the nodule. And so that low oxygen may have driven the preference for C4 dicarboxylates. We're not saying it's the only reason that the C4 dicarboxylates are used by the bacteria, but that may be one of the driving factors. So with that, I'll finish off by summarizing that we've developed what we think is a pretty comprehensive representation of the metabolism of legume nodule, representing the different zones, both the developmental stages and the stage of nitrogen fixation. And metabolic modeling seems to be a pretty useful framework for understanding the rhizobium legume symbioses. And so, like we've seen here, we can generate some hypotheses that can be difficult to look at experimentally, like the relationship between oxygen and C-paired carboxylates. It can also be useful for generating quantitative hypotheses that we could eventually follow up experimentally. And so I'll just finish off by thanking everyone that was involved. So as I mentioned, most of this work was done during my time as a postdoc at the University of Florence with Marco Fondi and Alessia Mangoni. We worked with Thomas Fowle, who prepared the Metacago model, and CERC for funding. Thanks.
So thanks, George, for this presentation. It was really clear and enjoyable. So I would now like to open discussion. Are there any questions from the audience? Okay, so the first question, why is sucrose not being taken up because of oxygen limitation in the root? Does the root keep the sucrose for itself?
Right, so the bacteroid is still getting the carbon it needs. So it's not that the plant is, say, keeping the carbon for itself, but instead of providing it in the form of sucrose, it's converting it first to C-paired carboxylates, and then providing that to the bacteria. So you can see that, yeah, the total amount of carbon provided is probably pretty similar. Why exactly that's happening, we never really looked into the specifics of it, but I think it has to do with the energetics of the usage of the different carbon sources. So since we're limiting the oxygen or the ability to produce ATP in the mitochondria, it seems to favor, well, not limiting it in the bacteroid, so the bacteroid can still use oxygen. It just seems to favor the switch in the carbon source, but I don't have a clear biological reason for that.
Okay. I've actually got a burning question. So you talked about the different zones in the nodules and how you model them by integrating transcript data. But what you didn't really show in your presentation are results. So how much do these zones differ metabolically, and how much would your modeling results actually differ if you were to develop an easier model that maybe only has like one general symbiotic interaction? So how much does it really affect your results?
Right. So yeah, I did leave that data out, but so these heat maps, the left is for Esmalodi, the right is for Metacago. I grouped the three differentiation zones together because they seemed pretty similar. So there wasn't much difference between like zone 2 distal, zone 2 proximal, and interzone. Well, essentially the gray is inactive, dark blue was essential reactions, and light blue were ones that were active in that zone, but non-essential. So there is variation between them. It wasn't massive variation. I think, say, in this region, there was an increase in reactions related to energy metabolism, perhaps to support the infection by the bacteria. There's more like carbon metabolism and nitrogen assimilation in zone 2, or zone 3, where the nitrogen fixation is occurring. I think overall the predictions at the growth scale would not have differed much if we just had a nitrogen fixation zone and instead added some sort of cost for the other zones which aren't fixing nitrogen but are using resources. So I think our overall results, as long as we accounted for that cost of supporting them, would have been similar. But one of the reasons for doing it this way was because one limitation of experimental work with studying the symbiosis is, say, you can delete a gene in the rhizobium and show it can no longer fix nitrogen, but you can't really tell why, what stage of the symbiosis is it blocked. So this was one way of letting us try and predict at what stage of the symbiosis might different genes be important, which you wouldn't be able to do if you only had the nitrogen fixation zone and represented the rest by costs.
Okay, thank you. Are there more questions from the audience? Okay, if this is not the case, maybe I will continue Okay, there are more questions. Sorry, too curious. In what form for nitrogen get to root amino acid? If the root is donating malate and taking back aspartate and asparagine, is it possible that the root is cheaping out on the symbiote by only donating a NADH equivalent?
Right. So,
Hope you got the question.
so yeah, so I'll first start by, just to clarify, the root will give sucrose to the nodule, and then within the plant nodule tissue, the sucrose is converted to succinate that is then sent to the bacteroid. The bacteroid then gives ammonia to the plant cells of the nodule. Within the nodule plant cells, the ammonia is incorporated into amino acids and sent back to the root as either asparagine or glutamine. I think in a model it went as asparagine. That does differ by the symbiosis, though. In soybean, it's ureides instead of the amino acids, but in this type of symbiosis, it's asparagine and glutamine. In terms of whether it's cheaping out, I mean, I think, so the plant wants the bacteria to fix nitrogen. So these bacteria aren't growing anymore. So I don't think it's so much that the plants have other methods of controlling bacteria. So the bacteria have limited cheating once in this stage, although there is some cheating. But so the plants do want the bacteria to have the energy they need to fix the nitrogen. I think it's probably more, yeah, I think it has to do with where the oxygens, or where the oxygen's available to the different organisms. So they want the bacteroids to use up the oxygen at high rates, and those enzymes are able to, while the plant is not able to. So yeah, I don't know if I'd necessarily call it cheaping out, but it is interesting why it switches to succinate.
Okay, thanks. Let's have one more question from Shyam. 30% of photosynthate is sent to nodules. Won't this limit other plants that are being engineered for nitrogen fixation?
Right. So yeah. So I'll answer that in two parts. The first is generally nitrogen fixation is an energy-intensive process, and so what plants relying on nitrogen fixation do tend to go slower than those provided like a nitrogen fertilizer. Although that does vary depending on the strain of the rhizobia as well. But one thing we didn't account for that could influence results is we didn't account for any root exudates or other sorts of like carbon sources that plants normally just throw into the soil. And so it's possible that some of that root exudate may be decreased to account for the increased use of it by the nodule. So it may not be, so yeah, so instead of say straddling however much photosynthate into root exudates, that might be decreased and reallocated. So it's possible that it wouldn't be as dramatic as seen in the simulations here.
Okay, thank you very much. So I think for me, it's now time to close the session, and I would like to thank again all the speakers. And I think this was a really nice walk on the frontier of what we're currently able to do with FBA and plant systems. So I hope you all enjoyed the presentations as much as I did. And having this said, yeah, I would like to thank the speakers again and then hand back to Shyam. Thank you.
Thank you very much, George, Nadine, and all the speakers for the day one. So I would like to request George to give away some takeaways from day one. Give his comments, please. George, if you can turn on, yeah.
I think, yes. Okay. Well, there's a nice symmetry about the day, starting with a George and ending with a talk from another George. Well, I think what I'd like to point out is that I think about 15, 16 years ago, in a review that I wrote with Yaya Shaker-Hill, we felt that it was still not going to be possible at that point to make genome scale models of plants that could be used for this kind of analysis. And then in about 2009, I think it was, Lee Sweetlove published the first FBA analysis of a plant metabolic network. And it was not apparent at that point just how powerful it was going to be. And I think what we've seen with the talks today starting with Nadine and running all the way through to George DiCenzo, is just how powerful flux balance analysis is. What it hinges on, of course, is accurate information about networks, but it also hinges on the ingenuity and imagination of the people who are pushing this technique forward. So I think we've had a really good demonstration of that, a whole series of demonstrations all through the day. And I have certainly learned a lot from listening to these speakers. I've nothing else to add, really. It's been a really enthralling set of talks. So I'm very pleased. Thank you. So back to you, Shayan.
Yeah, thank you, George. And thanks again to each and everyone who participated today. And on the slide, you can see that what is in store for tomorrow. Tomorrow's theme is 13C metabolic flux analysis in plant and microbial systems. So that would be very well complement. It can complement the flux balance analysis. And again, we do have a series of speakers as well as a couple of sessions where we'll discuss about the plant and microbial systems and the current challenges which we are encountering in the whole process, and which can also address some scientifically curious questions. So yes, thanks everyone. So we log off now. We close the session, close the day's session, and then I welcome everyone again to join tomorrow at the same time as today. Yep, thank you. Thank you, everybody. See you. Good night. We are ending the session now.
Thank you, Shayan.
Thank you.
Thank you, everyone.
Thank you, everyone, for the interesting talks.
Thanks to all the participants. And
5 minute है पकड़े.
yeah, Chandrakant, you are the host, so you can end the event.